So with those guidelines, let me introduce our IDRI fellow, Dr. Casey Youngflesh. He's a quantitative ecologist and a postdoc in Tingley Lab in the Department of Ecology and Evolutionary Biology. Dr. Youngflesh earned his PhD from Stony Brook University in 2018. He also received a NASA graduate fellowship over there. He worked at University of Connecticut before joining UCLA. His research interests are in addressing questions related to the timing of seasonal ecological events, trophic interactions, and population dynamics. His cross-disciplinary approach employs statistical and computational tools to integrate large-scale data from a variety of sources, including citizen science projects, satellite sensors, remote camera networks, and field-based efforts. He uses a range of methodological approaches, including hierarchical Bayesian models and quantitative remote sensing, to address both basic and applied questions in ecology. He will be taking over the first part of today's workshop on Introduction to Hierarchical Bayesian Modeling using STAN. So please join me welcoming Dr. Youngflesh.
Great, thanks, TV. Great, appreciate it. Yeah, so let me see if I can share my screen here. Does that look okay to you?
Yes.
Are you seeing the presentation?
That's right, yes.
Perfect. All right, well, welcome to the workshop. Thanks for everyone for coming. Hierarchical Bayesian Modeling with Applications for Spatial Environmental Data Science. So let's just quickly go through the schedule here. So the first bit will be an introduction to hierarchical Bayesian modeling using STAN. That'll be instructed by myself, as TV said. It'll be about a 3-hour session. We'll have a 1-hour lunch break from noon to 1. And then the afternoon session will be taught by Sudipto Banerjee, a professor in the Biostatistics department. And that section will be more focused on spatial data science using hierarchical Bayesian modeling. So kind of split into 2 sections here, each 3 hours long. 3 hours certainly is a long time to, have someone talk at you on a screen. So we'll try to break this up with a few breaks if we can. Yeah, we'll see what we can do there. As TV said, if you have a question, feel free to put it into the chat. I'll try to answer those as we go along. It might be a little tricky to monitor the chat and present here. So, do the best we can, especially in the breaks, try to come back to those questions. But yeah, you can drop questions in there. Few more logistics here. So a lot of this material will be sort of language agnostic. My hope is that you'll still get a lot out of this, even if you're not an R user, you're not a STAN user, any of that. That said, if you do want to follow along, there are several required pieces of software here for the first session. RStudio, R, and the RSTAN package are sort of absolutely necessary if you want to follow along on your own. Just go over to the course GitHub page. That's the link down there. Let me drop into the chat though. So just to make it a little bit easier here. Okay, that is the link. See if I can just bring up the page. So this is the GitHub page here. So all the information is in this README. If you're not familiar with Git or GitHub, that's fine. Don't worry about it. All the information is in here. If you scroll down to sort of this before the workshop begins section, this has all the required pieces of software in it. So it tells you exactly what you need to install for the afternoon session as well. We just put this up here this morning. So yeah, before the afternoon session, if you do want to, have some interaction there, go ahead and make sure you have all these things installed. Instructions are there. Code that I'll be using for this morning session, that there's hyperlinks to all of these here. So you can go ahead and, click on that. It'll bring you to the code. You could fork or clone this repository if you're familiar with Git. If those words don't mean anything to you, you can just copy and paste and, paste this right into your R terminal if you want. Or you can just code up in your R terminal, independent of this if you like. But all the scripts are here if you want to use those to follow along. The materials for the afternoon session are also in there. The presentations for the afternoon session are already up here on the page, and I'll put the PDFs of this presentation up for the morning session. I'll put this up here on this page this afternoon too. So probably within about an hour of us finishing, I'll put all that up there. So yeah, the GitHub page is really sort of the home base for all these different materials that we might need for this session. One other note, I'll try to put up the script name that sort of goes along with any of the sort of demos that we're doing here. I'll try to put that in the bottom right corner of the screen here, so you can follow along on your own if you want, if you don't want to be live coding on your own, if you just want to follow along. So look out for those little links in the bottom left here. All right, so this workshop is meant to give you sort of the tools to go out there and analyze your data, to further your intellectual journey in Bayesian analysis, if you will. You're not going to be an expert in Bayes in 6 hours, of course. There's really a lot of material that we'll present here, and you're probably not going to be able to digest everything, depending on sort of where you're coming from, your comfort level with some of these concepts. But really, overall, we have 3 major goals here. Firstly, to understand the basics of a principled approach to spatial environmental data science using a hierarchical Bayesian approach. Secondly, to fit basic models using STAN and R. And thirdly, to develop a base of knowledge that will facilitate future learning. So my hope here is that folks will kind of leave this workshop, ticking off all these 3 goals. Really, my hope is that regardless of where you're at, you'll take away something from this. And, in fact, my sort of money back guarantee here is that you'll leave this workshop knowing more than when you came in. And again, I'll post all the slides up for the morning session on the GitHub page. So I encourage you to sort of come back to these slides as we go along to sort of refresh your memory and come back to concepts that maybe didn't make so much sense the first time around. So a little bit about me, sort of TV already sort of gave an introduction here, but just to give you an idea about sort of where I'm coming from and how I might use these tools. One thing that I'm very interested in is sort of integrating a large number of data streams to sort of understand population community level processes, how these sorts of things vary over space and time. Hierarchical Bayes has really been crucial to my own work, sort of making it possible to model a whole host of different things using different data resources. And my hope here is that you'll find these concepts, these approaches as interesting and as useful as I have for my own work. All right, so what is a model anyway? What I'm going to do here, I'm going to ask folks to go ahead and think about this for 20 seconds and put into the chat, what is a model to you? When you're building a model to analyze your data, what does that mean to you? So just take about 20 seconds, throw into the chat. Yeah, what do you think a model is? Just kind of curious here. No wrong answers. All right, approximation. That it's a lot of that here. Find patterns in the data. Predicting output from inputs, a statistical representation, expression of relationships, mathematical expressions. Yeah, I think these are all great answers here. Relationships between inputs and outputs. Great. Yeah, so the first thing that comes to my mind is that, a model is some abstraction of some process of interest there. There's some particular way in which the world works, some processes that give rise to what we observe, and we're trying to simplify it to actually understand it, to represent it with a set of equations in our case. And really, it's this idealized representation of the data generating process. I think that's sort of one way to think about it, and one sort of helpful way that I like to think about this. And this idealized is really, and data generating process, sort of the key words here. So our model is not the truth. So it's important to keep this in mind when we're creating, when we're evaluating, we're trying to interpret our models. This is a quote from the statistician George Box, often quoted, that goes something like, all models are approximations. Essentially, all models are wrong, but some are useful. However, the approximate nature of the model must always be borne in mind. So we're not going to be able to represent the whole truth here, but that doesn't mean we can't gain some insight. So our goal is to use these models to advance the state of knowledge in our field. That's what we're doing here. We have our scientific questions of interest. We have our theories. We've made some observations out in the field or in the lab. We build our models, these idealized representations of the processes of interest. And then we use these to gain new insight, sort of about these things that we're interested in, as well as our uncertainty about this insight. And this ultimately advances knowledge, our knowledge as a field. This is what we're doing as scientists. So models really have this key link between our scientific questions here and sort of developing coming to some new insight here. Okay, so why use a Bayesian approach? Certainly we can build models without using Bayes. So why bother? It might be easier to not even bother with the Bayesian approach. And I really think the best answer to that question is flexibility. Folks might say different things here, but I think flexibility is really the key thing. We really want to free ourselves from the constraints of classical statistical approaches, where we might need to assume certain distributional assumptions. In some cases, I think you almost need a massive flow chart just to determine which statistical test you need to use. So we don't want to cram our question of interest and our data into some canned approach. We want to be able to build a model that suits our needs. And that's really what a Bayesian approach will allow us to do. That's what this is really all about, as far as I'm concerned. So there are a number of things that Bayesian analysis really excels at that I think are quite difficult in a frequentist or maximum likelihood framework. One of these is integrating multiple data sources, multiple sources of uncertainty, missing data, unobserved quantities. So by unobserved quantities, I mean sort of latent states that we might be interested in. We observe something, we might go out and count a number of birds, but what we're actually interested in is the true number of birds. And so we want to model that latent state. That's something we can do in a Bayesian framework, as well as forecasting, trying to predict how things are going to respond into the future, conditional on some set of future conditions. So I think all of these things really, Bayesian analysis excels at these things, and they're kind of tricky things to do in many other frameworks. All right, well, that sounds pretty good, but what the heck is a Bayesian approach anyway? What makes something Bayesian? Some folks that might be tempted to say, well, it's the use of prior information if you know something about the use of priors in Bayesian analysis. But that's actually not quite right. We can actually integrate prior information using some weighted maximum likelihood approaches, things like that. Really, to me, and to a lot of folks have written quite a lot about this, the Bayesian worldview separates things into two different bins. We have observed quantities and we have unobserved quantities. Observed quantities are going to be the data we go out there and collect in the lab or in the field, sort of what we actually observe, of course, what our instruments observe about the natural world. And our unobserved quantities are going to be things like parameters in our model. They're going to be these latent states, as we mentioned before. It's going to be missing data. So all these might be unobserved quantities. And in the Bayesian worldview, we seek to understand the probability distributions that underlie these unobserved quantities. So all of these things, these parameters, latent states, missing data, these are all governed by probability distributions. So we're thinking probabilistically here. And I think this is really the key component of the Bayesian worldview. So really what we're seeking to do with our analyses is to understand the probability distribution of those unobserved quantities given our observed quantities. So here, this PR here will just be probability of. This theta here is going to represent all of our unobserved quantities. So this could be, our parameters, latent states, missing data, what have you. This vertical bar here means given or conditional on. And our Y here is just going to represent all of our observed quantities. So all of the data that we actually observe, that we go out there and collect in the field, in the lab. All right, so this is really what Bayesian analysis is all about. Probability of the unobserved stuff given the observed stuff. Well, now you might be asking yourself, well, what the heck is a probability distribution? Really, a probability distribution is a mathematical function that gives the probabilities of occurrence of different possible outcomes. That's sort of the Wikipedia definition, and I think it's a pretty good one. So we have all of these different possible values of X here. And each of these possible values has a probability density associated with it. So this red curve represents all possible values for X and how probable they are. So this value would be more probable than this one over here in the tails of that distribution. So this is a continuous curve here, this particular one. The y-axis is just a probability density, and the area under this curve is 1. So that's one thing about a probability distribution. It integrates to 1 here. And these are used a lot in Bayesian statistics. They might be used to fit models to the data. These distributions are used to represent uncertainty in our parameters, and they're used to portray prior information, as we'll see here in just a little bit. So certainly used a lot, these distributions. I think you'll get used to thinking about quantities as probability distributions sort of as we go on today and as you sort of further your Bayesian journey here. So the shape of this curve that we had in the previous slide here, sort of arbitrary thing, that's mathematically described by the probability density function, often termed the PDF. So given mu and sigma here, the two parameters of the normal distribution, we can calculate the probability density at a given value of X. So this equation is the probability density function for the normal distribution. So that curve on the previous slide was a normal distribution. That this is the equation that sort of governs what that looks like. Now, of course, this equation doesn't look particularly fun to work with, and it's not. Luckily, we don't really need to worry about these equations for the most part when we're building our models. In fact, if we want to actually calculate the probability density for a given set of values, there's an R function to do that. We can sort of visualize this using a bit of R code. So this comes from 1-PDF in the GitHub repository, just to let you know if you're working through this on your own or you want to follow along. So here we're going to specify two objects here, mu and sigma, values 4 and 2. We're going to create a vector of x's from -5 to 15. So it's just going to be a vector of 1,000 values from -5 to 15 here. Then we're going to use this function dnorm in R. This is the density function for the normal distribution. We're going to feed it in this vector of x's we have. This is just arbitrary possible value of x's here. And then we're going to give it the two parameters of the normal distribution, this mu and this sigma, which correspond to the mean and the standard deviation here. And this is going to spit out a vector of probability densities at these different values of x. So this is a vector of length 1,000. Okay, so now we can plot those data here. So again, on the y-axis here, we have those probability densities. On the x-axis, we have these different values for x. And this is how the probability density changes over these x values, given a mu of 4, a mean of 4, which is this dashed red line here, and a standard deviation of 2, sigma equals 2. So basically, we plugged these values, mu and sigma, into this equation here. So we have 4 here, 2 here, 2 here, and these different values of x. And whatever this is spits out, that is the probability density. That is what's on this y-axis here. And so this area under the curve here is equal to 1 since it's a probability distribution. So here we have the normal distribution on the left here and the gamma distribution on the right. As we just said, and as most of you probably know, the normal distribution has two parameters, mu and sigma, that just so happen to be the mean and standard deviation here. It's Worth noting that this is just an interesting property of the normal distribution, that its two parameters are the mean and standard deviation, or variance, which is a standard deviation squared. That's not necessarily the case for all, or really any, most other distributions. So we can think of these two parameters as two knobs here to control what this distribution looks like. So we can sort of move these knobs around to change the shape of these curves. Of course, if we turn that mu knob to the left, the mean, the center of mass is going to move to the left, as if this, this green line here is a mu of -2. And then this sigma here, we can turn that knob and control how spread out that distribution is. So this yellow line here is a sigma of 5. It's a much flatter curve than this blue line, which has a sigma of 0.2. All right, straightforward enough. Two knobs, pretty straightforward change of the shape of these distributions. So just like the normal, the gamma has an equation that describes what these lines look like. It's a little bit of an ugly equation, of course. And we have these two knobs, these two different parameters, the way we've parameterized it here, alpha and beta. And we can, tune these knobs, turn them and change the shape. So here, this pink line, alpha equals 3, beta equals 0.5, whereas this red line is alpha equals 1, beta equals 1. Problem is here for the beta, it's a little bit more difficult to interpret how exactly these two knobs are changing the shape of these curves. So with the normal, that's, it's pretty straightforward, the center of mass and then the spread here, the location and scale, so to speak. The gamma distribution, it's a little bit trickier, but that's okay. You don't really need to know how to work with these equations at all. Just know that these equations are the things that underlie these different distributions. They govern the shape of these distributions. Each distribution will have its own equation with its own parameters that when changed, when you turn these knobs, they're going to result in a different shape for these lines. And just one note here, for continuous distributions like the normal, like the gamma, we call these equations probability density functions, PDFs. But for discrete distributions, discrete probability distributions where you can only have whole numbers, we call these probability mass functions, PMFs. Same concept, just a different name if you encounter that in the future. All right, so I'm going to bring up this web page here just because I think it's kind of a cool thing. There are, of course, many distributions. This is a number of them here on this web page. I think it's a nice sort of visualization here. You can sort of highlight different distributions. So here we're on the normal distribution here. And it shows the relationship of the normal distribution to all of these other distributions. So one thing to keep in mind is that all of these things are interconnected. The normal is connected to the log normal here. The standard normal is connected to the chi-squared. Often these distributions are just special cases or generalizations of each other. So there's all these different interconnected relationships between all these different distributions. Now, a little bit overwhelming, of course, when you see all these distributions. There's dozens and dozens of them. But really, we only use a small handful of these with any sort of regularity. So you can go ahead and sort of explore this on your own. You can sort of click through. It'll give you the properties of the distributions, which I think it's kind of interesting, but just kind of wanted to give you a sense of all the different options there are for these different distributions. So this is just a small sort of sample, small collection of distributions that we might commonly use for modeling things. This is certainly not an exhaustive list, but these are among the most common. And the distribution that you want to use is certainly going to depend on your problem. So the normal distribution, everyone's familiar with the normal, I think. The support of the normal is over all real numbers. You can have negative infinity to infinity there. You don't need to have integers, all real numbers. We have the log normal distribution. So that's non-negative real numbers. You can have negative values in the log normal distribution. The gamma distribution, same thing, non-negative real numbers. The beta distribution, that's a distribution with support from 0 to 1, all real numbers. So there are cases where we might want to bound our probability distribution between 0 and 1. Binomial distribution, that is a discrete distribution, so counts in two categories. So we'll talk about that a little later. The Bernoulli distribution, that's just a 0 or 1. So think about a coin flip. Might use a Bernoulli distribution. And a Poisson distribution, also very commonly used for counts. So that is a discrete distribution. If you're going out there and counting the number of birds that you observe or something, Poisson distribution might be appropriate. So here are a collection of scenarios here. Take a minute and think about which distribution you might use in each of these scenarios and why you would use that distribution. No need to put into the chat there. Just think about all these. We can do the first one here just to kick us off. It says presence or absence of a species in a collection of a forest. What might we use for this particular one? Well, if it's presence or absence, maybe we'd use a Bernoulli distribution, 0 or 1, depending on how we modeled it. So yeah, go ahead and go through these other examples just on your own. Take about a minute or two, and we'll come back and we'll discuss these. Give about 30 seconds. Okay, let's just quickly run through these. Here we have the number of plants in a 1 kilometer square patch. Well, number of plants, something that should come out is Poisson. It's a count, so that might be an appropriate choice here for number 2. For number 3, the mean summer temperatures across North America. Well, summer temperatures, they could take real numbers, really. They could be positive or negative, depending on how we sort of scale things. So we might use the normal distribution for that one, could be an option. The mass of adult birds across their range. Well, this one maybe a little bit trickier. You could use a normal distribution. You might want to use a log normal distribution. Mass can't be negative, of course, and when you're modeling mass, you're often sort of logging that just because that tends to be how it's distributed. So maybe a normal, maybe a log normal here. Total above ground biomass in a 1 kilometer patch. So this has to be positive. We can't have negative biomass here. So we might use something like a log normal or a gamma. So a gamma is a very flexible distribution that we might use here, or a log normal might be appropriate. And finally, here we have the probability that an individual survives to adulthood. So the key word here is probability. Probabilities have to be bounded between 0 and 1. So a good distribution to model probability might be a beta distribution because that has support from 0 to 1, any value in between those. So yeah, just sort of a quick exercise. So question in the chat here, what's the difference between the log normal and gamma? Both seems to require non-negative real numbers. Yes, they do, but they are shaped a bit differently. So you can think of the log normal as, well, let's say you've generated a number of random values from a normal distribution, you then log those values. If you were to histogram those, then they would be distributed log normally. The gamma is a little bit more flexible in that its shape can sort of conform to a bunch of different shapes. So, the exact choice, like why you make certain choices, that might depend on a number of different things. So yeah, it can depend, but Generally, the gamma is a little bit more flexible, but maybe a little bit more of a pain to work with at times. But they do have the same support. Okay, so Bayes' rule. All of Bayesian statistics is built upon this principle known as Bayes' rule, named after the Reverend Thomas Bayes, who came up with this idea quite a long time ago, actually, a couple hundred years ago. So we want to know what the probability of our unobserved quantities conditional on our observed quantities. That's what we said before. Bayes' rule is going to tell us how to get here, basically. Maybe we want to estimate the relationship between the size or weight of birds in a particular area and the amount of food there. We've gone and weighed these birds, measured the food, and we want to know what the intercept and slope of that relationship is between food and bird weight. So we want to know intercept and slope here are unobserved, and we have our data. So the likelihood here plays a key role. It is what we're using to link the observed quantities, our observed data here, to the unobserved theta. So it's saying, what is the probability of the data given a particular set of parameters? So this is the likelihood, and we'll come back to this again and again through the morning session today. And we calculate the joint likelihood as the product of the likelihoods of each individual data point, which we get using the density function. We won't go into too much detail about that right now. The prior distribution then represents what we know about theta, the unobserved quantities, before observing our data. So this might reflect some prior information that we have, maybe the results from a previous study, or maybe just what we think is biologically feasible. As domain experts, we know sort of what makes sense and what doesn't. So I think that's really an important key part of the prior here, feasibility. The product of the likelihood and the prior here is then proportional to the probability of our unobserved quantities given our observed quantities. So this little sideways dealy here means proportional to, and this quantity over here on the left is referred to as the posterior. This is what we're interested in, and this is proportional to the likelihood times the prior. So this here is known as Bayes' rule. Again, the theoretical foundation of Bayesian statistics here. You're probably saying to yourself, though, well, how did we get here? And that's a perfectly fair question. It all comes back to conditional probability. So we use the rules of probability to arrive at this statement. So let's say we have two events here. We have A and B, A in the red circle, B in the blue circle. The intersection of these two events is their joint probability, the probability of A and B. That's what this statement means here. So that's this intersection. They're both occurring. Now, there are two ways to think about this joint probability. We could think of it as the probability of B given A times the probability of A. So given the red here, how probable is blue? So it's right here, times the probability that A is even there in the first place. Or we could think of it as the probability of A given B times the probability of B. So sort of the same thing over here. It's just the inverse. Now, if A and B are independent, this intersection simplifies to the probability of A times the probability of B. So that's sort of a special case here. For example, if you had two coin flips, those are going to be independent probabilities. They don't depend on one another. So you could just multiply the probability of getting heads for the first, by the probability of getting heads for the second, to get that. But that's a special case here. We're not really necessarily assuming independence here. So this equation here is sort of this basic rule of probability, which may not seem immediately intuitive. I think it's something a little bit tricky to wrap your head around. But for now, I'm asking you to sort of trust me that this is true. We're going to move forward just for the sake of time here. Plenty of really interesting examples and resources online that might help you sort of intuit this a little bit more, though. So we've discussed these conditional probability relationships here, these conditional relationships. Now let's do some rearranging. Let's say that this over here, probability of A given B, is what we're really interested in. So we want to solve for this term on the far left here. Well, we can do a little bit of rearranging by dividing both sides by probability of B here. So we're left with just probability of A given B on the left. And we have probability of A and B over probability here on the right. All right. Now we know that probability of A and B is equal to this statement over here, probability of B given A times probability of A. So we can substitute this in for this term here, and we're left with this statement at the bottom, probability of A given B is equal to probability of B given A times probability of A over probability of B. All right. Now we've been talking in terms of A and B here, but the exact same thing applies if we're to be talking about theta and y, our unobserved and observed variables here. So if we just substitute these in, we have the probability of our unobserved given our observed, if we wanted to get, is equal to, this is the likelihood, probability of the observed given the unobserved, times the probability of unobserved, excuse me. This is the prior here. And this is, well, let's just flip forward for a second. The left here is our posterior distribution, what we're interested in again. Yes, the likelihood here on the top multiplied by the prior probability of the unobserved. And then in the denominator here, we have the probability of the observed here. And this is a little bit of an odd concept, I think. It's the probability of the data. Now, we often ignore the denominator here as the probability of the data. It's essentially this normalizing constant that changes the scale. So since we're interested in the relative importance of a given value, sort of in relation to all the other values, it can be ignored for our purposes here in fitting our models. So this will be a little bit more apparent, I think, when we talk about Markov chain Monte Carlo, MCMC, a bit later. So we're left with this statement on the bottom here if we ignore this probability of the data thing. This is why we say it's proportional to, because this is just a scalar, it's just a constant here. So we have the posterior is proportional to likelihood times the prior. If nothing over the last 5 minutes made any sense to you, that's okay, as long as you understand this statement, posterior proportional to likelihood times prior. This is a basic tenet of Bayes, and it's important to understand this at the bottom. All right, let's run through an example to help us better understand this. This example is from Richard McElroy's Statistical Rethinking. I'll talk a little bit about some resources at the very end of the morning session. I think this is a really awesome resource. We'll come back to that. But anyway, the question here is, what proportion of the Earth is covered with water? Maybe we're interested in estimating that. Now, let's say we make a number of observations all over the Earth, each time determining whether the location that we observed, so random location, is covered with water or land. So it's as if we have this inflatable globe. We toss it into the air and spin it around, catch that globe, look under our right thumb, and see if that location is water or land. Or any other sampling scheme that you might come up with. We just need to say water or land at a number of random points across the globe. So let's say that we toss this globe up and catch it 9 times, each time recording land or water. Now, if we wanted to answer our question with these observations, which probability distribution might be appropriate for that? Well, we have counts here. But we have counts in 2 different categories here. So because of that, we might want to use the binomial distribution. So here, W is going to be the number of times we get water. So it's the number of successes, if you want to think of it that way. N is the total number of trials here, the total number of observations. So that would be number of waters plus the number of lands. And P here is the probability of a success, proportion of the Earth covered with water. So here, W and N are observed quantities. We've observed these by tossing our globe up in the air, so we know what these are. This tilde means distributed as, so we're using this tilde a lot, describes how the variable on the left is distributed. So W is distributed according to a binomial distribution here with N trials and probability of success P. That's how you'd read that statement there. This binomial statement here, that is the likelihood. And this P is our parameter. That's our unobserved quantity. So it needs a prior distribution. We need to tell the model how that quantity is distributed. So P is a probability. What probability distribution might we want to use for P? Take 10, 15 seconds, throw into the chat what distribution you think we should use for P here. It's a probability. Okay, we have beta here. People think beta. Everybody agree? Well, yeah, the beta would be a good choice. We also have uniform here. Yeah, so since P is a probability, it's bounded between 0 and 1. So whatever distribution we use, we need to make sure it fits within 0 and 1. So the beta is, as we talked about before, we're going to go ahead and say a uniform distribution, though. We'll talk about a beta a little bit later, and we'll bound that between 0 and 1. So we can, bound the uniform between whatever values we want to, but since we have a probability here, we'll make sure it stays between 0 and 1. But yeah, beta is also a good choice here. So this P again, our unobserved quantity, our proportion of water, and we're going to say this is distributed according to a uniform distribution, and this uniform is our prior here. So all unobserved quantities need to have something on the right-hand side. By that, I mean unobserved quantity, tilde or equal sign stuff on the right-hand side. So these unobserved quantities are governed by probability distributions. That's the Bayes worldview. So here, this P is a parameter, so it needs a prior. But if you have latent states or missing data, other things like that, they need to be having something on the right-hand side as well. They might need to have a prior associated with them. Okay. Also worth mentioning that parameters can be modeled as a function of something else also. So they won't necessarily have a prior, but they'll sort of always be something here on the right-hand side. And we'll look at that a little bit later when we get into hierarchical models. So now we have our likelihood here, our binomial, and we have our prior here, our uniform distribution. Remember that our posterior is proportional to our likelihood times our prior. So let's rewrite this in terms of probability statements. So we have our likelihood here, probability of water given N and P, that's probability of water. Our prior here is the probability of P. Let's just say our prior knowledge for P is flat here over 0 to 1. That is, the Earth is equally likely to be covered by any proportion of water. We know that's not the truth, but for the sake of the example here, we'll just use uniform from 0 to 1. Our posterior then, probability of P given W and N, proportional to this binomial, so our likelihood times our prior. All right, now let's run through this model. After quite a lot of talking about models, let's actually run one using a grid approximation. So here we're going to generate some observations and some potential parameter values for P. So just to note, this is the script 2-globe.r in the GitHub repo. So let's specify our W as 6 here. We're just going to have one W and one N here. So let's say we tossed our globe up 9 times and we got water for 6 of those times. So our N is our total number of trials, W is water. This PP vector, that's going to be just a number of values from 0 to 1, a number of possible values for P, this sort of grid, parameter grid that we're going to sort of search over. We could then calculate the likelihood here by using this d binom function in R, so the density function for the binomial distribution. We're feeding it our W and our N, these things that we know, and then calculating that over a bunch of possible values for P here from 0 to 1. So that's what this represents here, this density here, this likelihood really on the y-axis, and all possible values of P here. Yeah, that is our likelihood. Probability of our data given our parameter. We then calculate the densities for our prior using d unit for density function for the uniform distribution. So over a number of different values for P, and we're holding those parameters for the uniform distribution constant, so from 0 to 1. So this is our prior. So this integrates to 1. It's a probability density function. We then multiply the likelihood times the prior, and that's proportional to our posterior here. So that's what we're doing in this line of code. We have our likelihood, this PR capital W, times PRP, which is our prior. So this is sort of the unscaled posterior here. What I've gone and done is scaled this posterior to integrate to 1 over here. So it's a proper probability distribution. We're not going to worry about scaling this when we're using MCMC because that probability of the data is a constant. Again, it'll be a little bit more clear, I think, when we look at how that algorithm works. But we're scaling that just so that the area under here is 1. So the posterior here looks very similar to our likelihood. And that is because we used a flat prior here for P. The prior didn't really contain any information that contributed something to the posterior. We can change the prior though and see how that impacts our results, see how that impacts our posterior. So using a beta prior, many of you suggested in the chat to use a beta prior, so the density function for the beta distribution, d beta, do the same thing we did with the UNIF, but with beta. That's what this prior looks like. So this is a much more informative prior, we might say, compared to a uniform. Basically, we're saying that, well, the Earth is much more likely to have a larger proportion of water than a smaller proportion of water. We may or may not want to do that, but anyway, just to show you how this works. But a more informative prior, we see how that changes the posterior distribution. So if we flip back, the mass of that posterior is more to the left here, lower with the uniform. When we use that beta, it's being pulled over to the right by that prior information. So you can see how that posterior changes there. Okay, so now using an approach such as this, this grid approximation, it's incredibly inefficient. What we just did, this search to arrive there, we looked across all possible values for our parameters, our one parameter here, P, and that was fine for this example, but it's not going to scale well. Plus, it was a little bit of a pain. There are several different ways of fitting Bayesian models that folks commonly use, but by far the most common method to do this is to use MCMC, Markov Chain Monte Carlo. MCMC is essentially a way to arrive at our posterior distribution in an algorithmic way. So instead of searching over the entirety of the parameter space, all possible values of P as we used before, it attempts to more efficiently explore the most probable areas of that parameter space. So it's really the machinery that's going to allow us to fit our models. So the goals are to sample from the probability distribution without knowing its true set of values here. So we want to get the posterior distribution, probability of the unobserved given our observed, but we don't know what it looks like. And that's what this model fitting is all about. We want to know the probability of parameters given the data. And the posterior allows us to make inference about this probability. So MCMC essentially wanders around the joint posterior distribution, spending more time in areas of high probability. So it generates samples using the likelihood and the prior and decides which samples to keep and which to throw away. So if everything is set up correctly, the amount of time the algorithm spends in a particular area of the probability distribution is going to be proportional to the probability of these parameters. So the idea is to draw enough samples from this posterior to make sure we correctly understand what that probability distribution looks like. So to help us kind of visualize this a bit, let's see if we can bring this up. This is kind of a cool demonstration, I think, that someone made to help understand how MCMC works. I think this is actually quite helpful. There are a number of different algorithms that we might use to implement MCMC, and they're all sort of doing somewhat similar things. They're trying to find areas of the probability space that are more certain. So let's imagine here for a second that this gray surface, this is our probability space right here. This is our posterior probability distribution. This gray surface is what we want to get at, but it's unknown, and that's why we're using MCMC in the first place. So here, each axis on this horizontal plane here is a parameter. So let's say this is sort of a 2-parameter probability space here. And the height of this surface, that represents the probability of that parameter combination at any sort of place on this horizontal plane, with lower values being better. So this might be like the negative log probability distribution. So it's inverted, lower values are better, higher values are not as good. So we start our MCMC algorithm, our chain, at a particular value here on our surface. So let's say we start up here and we kind of explore what the surface looks like. The goal is to get down here to the base, areas of higher probability. And this algorithm is sort of meandering its way down slope, which is what we want to do. You know, we're going to start at random places on the surface. We don't know what it looks like. And it's sort of reconstructing the surface as it moves down. It's trying to get down to this area here. So since it doesn't know what this looks like, at each sort of jump that it makes here, each iteration, it needs to calculate what the height of this surface is. And then it decides where to move next based on that height. Now, there's a bit of randomness here in how the MCMC explores this space. That's where the Monte Carlo comes in the name, to make sure that it doesn't get stuck in any areas. So essentially what's going on here, it's sort of making a proposal at each one of these jumps, calculating what the height is at this proposed space, and then deciding to move to that location with some probability that's proportional to how much better that sort of proposed location is compared to where it currently is. Now, this is maybe an idealized case here. It's this nice bowl shape. You know, starts up here, it kind of wanders its way down. You can speed up the animation here. Yeah, it comes down pretty easily. It's not too tricky of a problem. There we go. It's sampling the bottom of that space here to reconstruct what this surface looks like. That's what we're trying to do. Probability space might be a little bit trickier, though. It might be sort of one of these sort of double bowl shapes, multimodal, you might say. This can be a little bit trickier. You can see here that the algorithm is kind of getting stuck in this first bowl over here. It's not quite jumping over this little ridge to the other bowl. If we were to look at the results of this exploration, we wouldn't see this space over here at all. Again, we're kind of trying to reconstruct what this looks like based on these samples. So the fact that this isn't exploring this bowl over here is definitely not a good thing. We want to know what the whole space looks like. So what we typically do here to help sort of address this problem is to use multiple chains. So multiple realizations of this exploration. So instead of this one chain exploring the probability space, we might use 3 or 4 started at sort of random locations across this probability surface and making sure they're exploring the same areas of the probability space. If they are exploring the same areas, that's great. If they're not, well, then we can't make any valid inference on our model. And we'll come back to that when we see, we'll go back to the diagnostics here. Now, different algorithms explore this space in different ways. Of course, many of these can sort of tune themselves to make sure they're exploring this parameter space efficiently, effectively. Some of the common algorithms used for MCMC include Gibbs sampling, Metropolis Hastings. Those are used by pieces of software such as Bugs, which I think Sudipto is going to use this afternoon. JAGS, Nibble, those all use Gibbs, Metropolis Hastings. You might have heard of some of those. Stan, the piece of software that we're going to use today for the morning session, that uses something called a Hamiltonian Monte Carlo. So that works a little bit differently than Gibbs or Metropolis Hastings. So the way you can think about Hamiltonian Monte Carlo, it's almost as if you have a ball rolling around on this probability surface. So imagine we have a ball that we're placing on the surface. We're going to give it a little flick. And that ball is going to roll around the surface, sort of reconstructing what that surface looks like as it goes along. Remember, the shape of this gray surface isn't known. That's the reason why we're doing this whole MCMC thing in the first place. So it's figuring out what this looks like as it goes along. And there are all sorts of clever ways that this explores the space and decides sort of where to go. But for today, I just want you to sort of get a very general sense of what's going on here. Hamiltonian Monte Carlo is like a ball rolling around this sort of n-dimensional probability surface. Again, this is sort of a 2-dimensional probability surface because we're really only able to visualize this in 2 parameter dimensions, the third dimension being probability. But of course, if you have a parameter with 1 million models, well, you're going to have a 1-million-dimensional space. So you can see how this gets kind of complicated quite quickly. So this demo has a few different options for probability, what that probability space looks like. We looked at the bowl here. We looked at this sort of thing right here, whatever you want to call that. Posterior could be a lot more complicated. You know, this is sort of this canyon-looking thing. So it might be a tricky thing for some algorithms to explore complicated probability distributions like this. Luckily, there are entire fields devoted to optimization and figuring out how to explore this space. And we're just going to sort of take advantage of that, which is excellent that we don't have to do that. So question in the chat here, how does it compare with gradient descent algorithms? Yeah, I know that there's a bunch of different algorithms to do gradient descent. Yeah, I'm not super familiar with a lot of them. Again, there's a bunch of different ways to do MCMC. Hamiltonian Monte Carlo is one, Gibbs sampling Metropolis Hastings is another one. Metropolis Hastings is more of like a random walk exploration, so it's less efficient. Hamiltonian Monte Carlo is kind of the cutting edge here as far as MCMC algorithms are concerned. Yeah, well, hopefully that was kind of helpful in visualizing what these things look like. Ultimately, the concept is very similar to gradient descent. You're trying to sort of find the best place of this distribution. So sort of a similar problem here, but a little bit different in a couple of different ways. So let's go ahead and take just a 10-minute break here, and we'll come back and work through an example. Hopefully the examples make it a little bit more clear, connect some of these concepts together. Yeah, let's come back at 10 after. Come back at 1:10 and we'll continue with some other things. All right, folks, let's get back to it. It's been about 10 minutes at least. Just a couple questions in the chats here, a couple comments. One comment here from Eric. I think gradient descent is designed to seek out local optima, whereas MCMC is designed to explore the parameter space around the optima. Yeah, it's definitely true that MCMC, we're not just trying to find the low point, we're trying to explore the area around that low point to try to get some idea of the uncertainty there. So we definitely want to sort of reconstruct that surface. That's actually what we're trying to get at. Although we are certainly trying to look for the global optimum rather than the local one here. We want to end up in the best place that we can find, that lowest space, and explore that space if we can possibly get there. There's another comment here, question. Is it fair to say MCMC is just generating random numbers to explore the optima? Well, it depends on which algorithm you're using. Since it's a Markov chain, it's actually sort of meandering its way through where its next position is dependent on where it currently is. So you can imagine, you're sort of a person like walking through this probability space. Your next step is going to be dependent on where you currently are. And there's a sort of a complicated, potentially complicated set of rules, decisions to be made based on a number of different things. Some of that is random, and some of that is, the characteristics of what that probability surface looks like around you. And exactly what those rules are is going to depend on the algorithm that you're using. So Metropolis Hastings, for instance, is largely a random walk. You're in a place, you pick a proposal destination, like, oh yeah, over there. You calculate the probability at that spot, and then you decide to move there with some probability. There's some randomness there based on whether that's a better location than you currently are at or not. So it just depends on the algorithm here. I don't want to get too bogged down on MCMC because it's kind of this machinery that we're using, but that'll sort of give you a general sense of what's going on here. Okay, so let's go through another example here. Let's say that we're interested in the effect that bird food abundance has on the weight of a particular bird species of interest. So here, let's say Y is bird weight, and X is bird food abundance, where these little black things are caterpillars. Let's just say this particular species of bird eats caterpillars. We have this arrow here to illustrate the direction of the effect here. We're interested in the effect of food on bird weight, not the other way around. So we think that maybe our food might give rise to heavier birds. More food might give rise to heavier birds. So sort of this thinking about this generatively data generating process here. And of course, we might call these two variables a number of different things. Sure, most of you have heard some of these terms before. These Y's here, we might call the dependent variable, the response variable, the outcome. These are all synonyms here. Whereas the X variable, we might call this the independent variable, the predictor variable, explanatory variable, covariate. There's probably some other terms as well. Certainly a number of synonyms here, but these are both observed quantities. We go out to nature and we weigh birds and we count caterpillars here. All right, well, before we do anything else for this problem, let's go ahead and simulate some data. Let's create some fake birds here. After all, we all know that birds aren't real anyway, right? So what's the harm in making up a few more? Joking here, of course, as if that needs to be stated. While birds are most certainly very real, simulating data, creating some fake data, is an excellent way of checking to make sure that our model obtains values similar to those that we use to actually simulate our data. So simulating some data, fitting that model to it, is a great way to make sure your model is working. In a simple linear regression case, maybe it's not really such a big deal, but once you start fitting more complicated models, it's always a good idea to make sure your model is sort of doing what you think it's doing. The best way to do that is to use simulated data. So fake birds are useful. So let's generate some data. This is 3birdsimulation.r, the script in the GitHub repo. Feel free to follow along in your own terminal, or just to go through the script if you like, or you can just watch the screen, whatever you feel is most useful for you. So the first step, what we're going to do here is set the seed. Just going to pick an arbitrary number here, set C1, just to make sure we can reproduce these results. Whenever you're generating random values in R, it's best to specify this seed to make sure you can reproduce the exact values. So we're just going to set the seed to 1. We're going to say that we're going to have 30 data points here. So we're going to set this variable n equal to 30. And I'll try to work through fairly slowly here so that folks working through on their own can have time to get through this. So do my best to sort of move slowly through here. Now we're going to simulate our predictor variables. This is going to be our food here. So here we're using R norm to generate random values from the normal distribution. We're going to generate 30 of them, n, with a mean of 0 and a standard deviation of 1 here. So here our variables are going to be standardized. So it's common to standardize your predictive variables to make them easier to interpret. So this is as if we went out and measured a bunch of caterpillars at a bunch of different sites here, and then standardized those values so the predictor was centered on 0 with a standard deviation of 1. So that way, a 1 unit change in our predictor variable represents a 1 standard deviation change in food abundance. So there's this nice interpretation there if we standardize this. So just simulating this as if it was standardized. We can then specify the relationship between bird food and bird weight here. We're going to make up some values for an intercept and a slope parameter here. So where our alpha is our intercept, our beta is our slope, just choosing values of 40 and 3 here. We then use these values that we made up in conjunction with our predictor variable, our food STD here, to simulate our linear predictor, our mu here. So the linear predictor is essentially the expected value of y for a given x according to our model. We'll come back to that in just a minute. So we have a vector of length 30 for food STD. Our mu is also a vector of length 30. We then specify the process error with regard to the relationship between bird food and bird weight. Again, we'll come back to this in just a second. So specify sigma is equal to 3. Then use R norm again to simulate some values for bird weight, where n is the number of samples once again. Mu is the mean and sigma is the standard deviation. So bird weight here, again, a vector of length 30. We can plot the simulated data now. So we have simulated bird weight on the y. We have bird food standardized here on the x. And just as a little bit of an aside, I think visualizing your data is among the best pieces of modeling advice out there. It might tell you things about the structure of your data that might inform your model building decisions. So always plot your data. A little bit of an aside there. All right. So these data in this plot shouldn't be too much of a surprise. We made these values up after all. There's a positive relationship between bird weight and bird food. Slopes about 3, intercepts about 40. Well, that checks out because we chose those values. So of course, we just simulated these data. So we know the true generating process here. But let's pretend for a moment that we didn't know that we've gone out there into the world, collected these data, don't really know too much about what we're going to get. Now, the relationship between bird food availability and bird weight, this might be very complex and nonlinear. But models, of course, are just an abstraction of reality. So to simplify all these intermediate processes that might be at play, all these different complexities, we might model that relationship between these two variables using a linear regression here. So this plot here might be our sort of a priori expectation, be my expectation at least, more bird food, heavier birds, although there might be a number of reasons why we might not see this pattern. So again, we have weight on the y, food on the x. let's write down a model here to try to fit these data. So for this model, what distribution might we use to model bird weight here? Just don't need to type it in the chat. Just think about it to yourself, 5 or 10 seconds. What distribution might you use to model bird weight and why? So I'm going to go with a normal distribution here. You might say, well, bird weight's positive only, so I'd like to use a gamma or a log normal. Log normal would be equivalent of logging your weight data and then modeling it as normal. So, maybe this is logged weight or something. Some of these decisions regarding what distribution is best to use, there's no one right decision. I'm going to use normal here for the sake of just sort of ease in going through this example. But certainly the normal is a very common distribution. So I'm going to go ahead and use that. All right. So why is our response variable here bird weight, where I is the observation? It is our index. So y is a vector of length 30. So we have 30 different i's here, 30 different observations, 30 different indices here. And each y has its own mu here. It has its own linear predictor. Mu is the expected value here. So again, referred to as the linear predictor sometimes. It describes the values along this red line, the values of y given a particular value in x. It is essentially the model's estimate of the mean behavior. Now each response variable here, it has its own mu, and each of these mu's has an x associated with it, a predictor associated with it. So y is of length 30, x is of length 30, mu is of length 30, where mu is equal to alpha plus beta times x, where alpha is the intercept term and beta is our slope here. Intercept, of course, where the line intersects the y-axis, and our slope is our rise over run, how steep that line is. Sigma then represents our residual error. So how much the data deviate from this line. Excuse me, I'm just going to take off these headphones. I think they're running low. All right. So if these points, the observed data here in this plot, are further away in general from this line, from our expectation here, the sigma is going to be larger. If they're closer to that line, the sigma is going to be smaller. So this sigma here is essentially the leftovers, or residual variation in the data that mu is not accounting for. So this is the general structure of our model. Take about 10, 15 seconds and just identify which of these quantities are observed and which are unobserved. No need to put it in the chat, just do this to yourself. 20 seconds, which of these are observed, which are unobserved. So in this case, our y's and our x's are observed, our response and predictor variables. These are what we measured in the field, our bird weight, our food abundance. Everything else is unobserved. We don't know what this intercept, this slope, and this residual error are. We're estimating those in our model. And just to note that these particular Greek letters that we use here, they're kind of the conventional ones that we use for these particular types of things, intercept, slope, and residual error here. But you could use anything. So you don't have to use an alpha for your intercept. You don't have to use your beta for your slope. We're just using it here because that's typically what is used, but it's entirely sort of arbitrary, really. All right. So remember that all of the unobserved quantities in our model are governed by probability distributions. That was our Bayesian worldview that we lined out. So these need to have something on the right-hand side. So mu has something on the right-hand side here. Mu is actually kind of this intermediate variable, since we could jam all of this alpha plus beta times x up into here. So mu is this sort of, intermediate, or even sort of intermediate quantity, let's call it that. Alpha, beta, and sigma, they don't have anything on the right-hand side now. They need prior distributions. So which distribution might we use for alpha, our intercept? So if we think back to all of our probability distributions, all the options, I'm going to go with a normal here. Pretty acceptable, pretty good options here. Normal can take all real values, could be negative, could be positive. That sounds like a good choice here for our intercept. What about beta, our slope here? I'm going to go with a normal for the beta as well, for the prior. Again, slope could be negative, could be positive. It could be, any real number here. So we'll use a normal distribution for the prior for beta. And then finally, we have sigma up here, our residual variation. What distribution might be appropriate here? Well, since it's our residual error, it's the standard deviation here, it has to be positive. We can't have a negative standard deviation here. So with STAN that uses a Hamiltonian Monte Carlo sampler, MCMC sampler, the half normal is actually a very good choice here. And it's actually recommended by some of the STAN developers. And you might be saying, well, half normal, I haven't really heard of that before. A half normal is just a normal distribution, the classic bell curve that's been truncated at 0. So it's been cut off. In this case, it's just the positive portion of that normal distribution. So that way, negative values are not possible for this parameter. That's what we want. So the gamma distribution has traditionally been a popular choice for the residual error. You'll likely see this in a lot of older statistical textbooks. Might even talk about it this afternoon, since Sudipto is using bugs rather than STAN. If you remember though, while gamma is non-negative real numbers, it has these two knobs that aren't very interpretable. So it's not ideal in that sense. It does happen to work very well in conjunction with the normal distribution for certain MCMC algorithms, namely Gibbs sampling. So there's something called conjugacy that we won't cover today, at least in the morning session. And the gamma is conjugate to the normal, which is our likelihood here. So really the history of using the gamma comes from some computational efficiency that you get with Gibbs sampling. Again, the knobs of that gamma there are not very interpretable, so it makes it difficult to set sort of intelligent priors here. And we want to set intelligent priors. So in STAN, we're benefiting from not using the gamma. So we're just going to go ahead and use the half normal here for the residual error. A good choice, easy to interpret the parameters for that half normal distribution. Okay, so we decided on which distributions to use for our priors, but now we need to specify the values for these distributions. We need to fill in all of these question marks that we're left with. Which value should we choose here? Well, it's important to think about what each of these parameters means, namely how to interpret them. So the alpha parameter here, that is our intercept. As the intercept, it is the model's estimate of bird weight at an X value of 0. So since we've normalized our predictor variable, which is food abundance here, 0 represents mean food abundance. So by normalizing that, alpha is bird weight at mean food abundance. So we might use what we know about birds to set this prior. So let's say we know that bird weight is, around 50 grams for these sorts of songbirds. Let's pretend we're all ornithologists, domain experts. We kind of have an idea. It should be around 50 grams. Not quite sure what it is, but maybe we use a normal distribution centered on 50. Then for the standard deviation of that distribution, maybe we use something like 15. So 2 standard deviations would be about 30. So that'd be a range of about 20 to 80 grams. That's a pretty big spread, aligns with what we know about the biology of this bird. We think that's biologically feasible for possible values of the weight of this bird. And just to note here that Stan uses the standard deviation here as the second parameter of the normal distribution, just like R does, same thing. So in my notation here, we'll always be assuming that the second parameter of the normal is the standard deviation rather than the variance, which is often used. So for beta here, the slope parameter that we have, that can be interpreted as the steepness of this line. So again, since the predictor variable is standardized, the interpretation is for a 1 standard deviation change in food abundance, weight changes beta amount. So how much would we expect bird weight to change with an increase in food? Well, maybe we center this beta, this normal distribution for beta, our prior on 0. Even though we expect beta to be positive, we'll set it to 0 here, center it on 0, and give it a reasonable spread for biologically reasonable values here. So with standard deviation of 10, 2 standard deviations for the prior would be from -20 to 20. A slope of 20 would mean a 20-gram change in weight for a 1 standard deviation change in food. That is quite a lot for a bird that weighs 50 grams. So definitely at the edge of what's biologically feasible, but we'll go ahead and use that. It's a nice sort of weakly informative prior, we might say, for our slope there for beta. And now what about sigma? Well, for this, we need to think about how far off our points might be from our line. Well, if weight is something like 50, again, these are pretty small birds, 50 grams, residual error of 20 would be quite a lot. If this value is 20 grams, we don't really expect it to be any more than that, even in the sort of very extreme cases. So maybe we set the mean to be 0 here for a half normal and the standard deviation to be maybe 10. All right, well, there we go. So what we did here is set priors within a range that we thought were biologically feasible here. So I think it's important to point out that there are no correct priors here, just scientifically justifiable priors. And it's really important to keep in mind that priors only make sense in the context of your model and your data. So reasonable priors for one model are not necessarily going to be reasonable priors for another model. It's important to think about what each of these parameters means and set your priors accordingly. So I think it's important to justify these priors with information outside of your data. So you don't want to figure out what your data looks like and then set your priors based on that. That's sort of like double dipping with your data. So priors should always be set based on outside or a priori knowledge or expectations. Now, there are other ways of choosing priors, things like prior predictive checks, sort of a principled way to do this. We're not going to go into that for the sake of time, but know those sorts of things are out there. We can also check to see how much of an effect our prior has on our posterior after we fit the model, sort of as a gut check to see if we made any mistakes or maybe we're sort of biasing our results in some way. But ultimately, at the end of the day, if you don't know much about the set of reasonable values for a parameter, and you might not many times, you just set that prior to be what we might call uninformative. So we just set a very diffuse prior that isn't contributing much knowledge. So it'll be something like the flat prior that we use, the uniform prior for P in our previous water earth example there. That's the sort of conservative approach there. So I think the takeaway here is really that priors need to be set on a problem by problem basis, and you do need to sort of think about them when you fit the model. Okay, let's write this model out. We've kind of written it out here, but let's put it in terms of probabilities here. Here we have the probability of our data given our unobserved quantities, our alpha, beta, and sigma here. This is our likelihood, this top statement. We then have the probability of our unobserved quantities, our probability of alpha, probability beta, probability sigma. These are our priors. And again, this mu is sort of this intermediate term. So it's sort of this intermediate quantity. It's not really a parameter in the model. So these things are really the most important bits here for our unobserved quantities. All right. So we have our likelihood here. We have our priors down here. The posterior then is the probability of unobserved stuff given observed stuff. It's sort of the inverse of the likelihood. Probability of our parameters, alpha, beta, and sigma here, given y and x, which is our observed bird weight and food abundance, these things that we've measured. And this is proportional to the likelihood, normal distribution here that we decided we were going to use, times the priors. And we have three different priors here, the prior for alpha, prior for beta, and the prior for sigma here. Now we could do the same thing we did before with the globe water land example here to fit this. We could use a grid approximation, choosing multiple values for each parameter, calculate the likelihood at each parameter combination, multiply that by the prior, scale that to get our posterior. But choosing just 100 values for each of these parameters would be 1 million different combinations. And this is an incredibly simple model. So that grid approximation here is really not a good approach, as we said before. It's not an efficient approach. So what do? Well, MCMC to the rescue here. We'll use Stan to run this MCMC machinery. We're not going to code it up ourselves. You know, people have done the hard work to develop these tools, and we're going to take advantage of that. So just to provide a bit of background here, Bayesian statistics has really only become widely used over the last several decades. And that's really to do with the computational nature of fitting these models. So we've come a long way, both in terms of the statistical and algorithmic machinery to fit these models, as well as the actual hardware to do the hard work that we need to, really just many advances over the last several decades in both of these things. It was really in 1990 where some research by statistician Alan Gelfand made it possible to fit Bayesian models in a tractable way. Of course, building off the work of a number of other folks. And now we're going to take advantage of all that to fit these models that we want to fit these flexible models. All right, well, let's open up RStudio and work on understanding how to actually implement these models, this particular model in Stan. Just bring up RStudio here. So I'm going to kind of live code this with you, for better or for worse. All right, so we've got our RStudio window open. If you want to follow along on your own, feel free, or just watch the screen, whatever you think is most informative for you. If we go to the upper left in our RStudio window, this is where we create new scripts, create new markdown documents, et cetera, et cetera. You'll notice that Stan file is one of the options here for new files. So go ahead and select Stan file here. We're going to create a file for our Stan model that we're going to use to fit our model. And it gives us this sort of template here. So while we interface with Stan through R, we need to write our model out in the Stan language. Stan has its own syntax. It's really pretty similar to what we've already been writing out, though, with our models. So it's pretty user-friendly. When we call our model in R, we then refer to this Stan file. So this file specifies our model here. And RStudio and Stan give us this example model here, example Stan file to get us started, which is pretty helpful. It's a starting point here. And there are some comments up here right at the top. So these double forward slashes, these denote lines that are commented out here. So just said this program defines a simple model with a vector of values y. Model is normally distributed with mean mu and standard deviation sigma. Okay, it's just kind of describing what's going on here. Go ahead and delete that from the top just to clean this up a little bit. All right. So Stan files are broken up into different blocks. Each of these are blocks here, bounded by these curly brackets. The first one here is the data block. This is where we define all of our data, all of our observed quantities that we're feeding into the model. So Stan is very explicit here and breaks things up into these different blocks. It's also very strict about how you specify everything in the model. So this might seem like a big pain at first, and you might be sort of cursing Stan that this is the fact, but it's actually so great when you get used to it. The fact that you need to be explicit will help you to debug your models when they inevitably fail. This is something that other pieces of software used to fit Bayesian models are not very good at. So this makes the debugging quite a lot better. So data block here, the first thing, we're declaring a object named n, and that is of type integer. As int is integer, and it is bounded at the low end by 0. And then we have the semicolon here at the end of the line. We need semicolons at the end of each Stan line to make it run. So again, very explicit. Don't forget your semicolons here. So we've declared this integer n. The next line here, we're declaring a vector y of length n. So we need to declare n before we declare y, because y is of length n. And we could have hard coded in, 30 here and got rid of this line, but in general, that's not good practice. We want our model to be, we want our Stan file to be fairly flexible if we change the number of observations we have in R. So best to, declare n here, declare y as a vector of length n. All right, so that's our data block here with our particular example that Stan just gave us, our studio gave us. The next block is the parameter blocks. We'll delete these columns. This says the parameter accepted by the model. Our model accepts 2 parameters, mu and sigma. Yeah, so this example model has 2 parameters here. So we've declared mu as type real, so it's a real number here, and sigma also of type real, so that's also a real number. So there's a question in the chat, where's x here? Yeah, so we haven't included x in our model. This is just the example model that RStudio gave us here. We're going to work on building up our model that we just wrote down here in a second. This is just a run through an example. Sigma here of type real, bounded on the lower end by 0. If we look down here in the model block, well, sigma is the standard deviation in this particular model, just like in our model. So it can't be negative. It's bounded at the lower end by 0. So yeah, we specify lower bound 0 here. We now have the model block, and this is where we actually specify our model. It says the model to be estimated. We model the output y to be normally distributed with mean mu and standard deviation sigma. So in our model block, we have y goes as normal, mean mu, standard deviation sigma, and we're closing it off with a semicolon there. So that should look very familiar to you. We're using that little tilde, just like we've been doing. So, once you've written down your model with a pen and paper or in a text editor or something else when you're working on your own, which I highly recommend, translating that to Stan is relatively painless for the most part. You're writing down your model just as you would. You're making sure you declare your unknowns and your knowns, your observes and your unobserves there. Okay, so this right here is going through every element of y, which is specified in the data block. The mu and sigma are unknowns. There are parameters here. And then remember that our unknowns are governed by probability distributions. So these parameters need something on the right-hand side. They might be being modeled as a function of something, but if they're not, well, they need to have priors. But priors appear to be missing here. Well, surely the Stan developers did not forget to add priors for their example model. Well, they did not. In Stan, if you don't declare a prior for a particular parameter, it gives that parameter a uniform prior from -infinity to infinity. It's what's called an improper prior. And we don't want to let Stan do that for us. We should always define our priors. So while Stan is usually very explicit, this is the one way it unfortunately is not. So let's explicitly give some priors to these parameters. So our mu here, well, let's just give mu a normal prior. Won't worry about the exact values right now. And let's give sigma a half normal prior, as we did with our model here. You might be saying, well, how do we give sigma a half normal prior? We need to put half normal in here. Well, since we declared sigma as bounded on the lower end by 0 here, we can actually just specify a normal prior for sigma. Sigma is going to automatically truncate this distribution at 0, so it's positive only. So this is a half normal prior, which is really convenient here. Just specify the normal and make sure you bound that there. So let's go ahead and just make a comment here. Double forward slash is the comment and stand. We'll just say half normal, so we remember in the future what this is. All right. Now, I just left the x's here because we don't have any data here. This is just an example model. Let's just say this is unrelated to the bird example that we've been talking about. If we don't know what our data are, we can't really set our priors. Maybe we could decide to set our mu here. Maybe the mean of the normal distribution is set it on 0. Maybe we say, oh, well, we want a standard deviation of 10. But remember, our priors need to be interpreted in light of our model and our data. And without some specific real or fake data, we can't really, it just doesn't make any sense. Maybe our data are, in the hundreds or in the thousands, and this prior, wouldn't make any sense there. We'll just change these back to X for now. Now, one incredibly useful thing about using RStudio, stand in RStudio, is that RStudio speaks the stand language. It will tell you if there's something that's not correct about your stand file. Now, there's no way to run this stand file line by line as you might run an R script line by line. This stand file gets passed to R in its entirety. But because RStudio speaks stand, you can debug certain things about your model without actually having to run it. So to do that, we just need to save this stand file. Just to start, I'm just going to call this temp here, just to save it. Doesn't really matter what we call it here. Just saving it so it'll evaluate our file to see if there are any problems here. What we can see here is that this little X here with this red circle, it says variable X does not exist. So it has a problem with this line 13 in our code. Well, we have this X here. We haven't declared X in the parameters or the data block. So we would need to do that if we're using something here. We want to put numbers here. We don't have data, but just for the sake of seeing how RStudio works with this, let's just put in some numbers for both mu and sigma here. We see those warnings go away. So what RStudio is telling us is that, hey, your stand file looks pretty good. It's not, unhappy about anything. So incredibly useful for catching typos in your model there, doing this in RStudio. So I highly recommend using RStudio to do this. All right, let's code up the model that we just wrote down, focused on the bird weight and bird food. This is that model. So I've saved the sort of final copy here, the completed stand file for fit_linear_bird_model.stand in the GitHub repo. So you can open up that if you like and just sort of go through it, or if you're doing it on your own, we'll work through and sort of live code this up either way. But this is the sort of finished file here, if you're curious now or later about that. Let's build off of this sort of template script that it gave us and implement this model that we wrote down. We're already sort of halfway there. Okay, so we've decided for our model here what our observed and unobserved variables are. Our observed variables were our y's and our x's, our bird weights and our food abundance. So we need to declare those in our data block. Well, right now, we just have y and n declared in our data blocks. We're missing our x. as the person in the chat pointed out. So let's declare x a variable, a vector of length n. So x is the same length as y. It's a length of 30 actually, since we generated the data. So we have our two observed variables there, our x's and our y's. Okay, let's go to the parameters block here. Well, we have our three parameters, our alpha, our beta, and our sigma. We have sigma here already. Alpha in there, and we'll put beta in here. So these are two parameters here. So we have alpha, beta, and sigma. Check, check, check. Okay. But now we have this new sort of intermediate object. What are we going to do about that? Well, the easiest thing to do is to add another block called transform parameters. And here we're going to declare mu as a vector here of length n. So it's the same length as y and x, as we see over here, same length here. So we're going to declare a vector, mu, a vector of length n. Then we're going to say mu equals alpha plus beta times x. So alpha plus beta times x. Remember our semicolon there. We can go ahead and save that. So again, we could have just omitted this mu and jammed all this up here into the normal, but this is just a little bit easier to keep track of. Plus, we're tracking our expected value or linear predictor. So a number of reasons that we can use mu here. Okay, we sort of ticked off the data, the parameters, and the transform parameters block. Now we're down here to the model block. We don't need a prior on mu in this case. There's stuff on the right-hand side of mu. It's this intermediate value, intermediate quantity, excuse me. We do need priors for these other parameters though. So we need a prior for alpha. So we said our prior was going to be a normal centered on 50 with a standard deviation 15. So we have normal here, centered on 50, standard deviation 15. Use a semicolon. And our prior for beta here, normal 0, 10. So beta, normal 0, 10. All right. There we go. We've set the priors for our parameters here. And then this final statement here, our likelihood, you can just add a comment here to keep track of these things if we like. Our likelihood is y goes as normal with mean mu, standard deviation sigma. Well, that's what we have here. So that actually stays the same. We don't actually need to do anything here. All right. So we've got ourselves a stand file here, it looks like. No errors that our studio has identified, which is good news. But we're going to actually do one more thing here. We're going to add one more block called generated quantities. All right, generated quantities. We're going to specify a new variable here called yrep, and this is going to be of length n. We're going to say that yrep is equal to normal RNG mu sigma, and with the semicolon. What this is going to do, this normal RNG, it's a random number generator. So all of this up here, this is actually to fit our model. This down here is just going to produce some stuff that's unrelated to actually fitting our model. We're going to use this to generate new data. So that's what this normal RNG is. It's going to say at each iteration, these parameter estimates, it's going to use them to generate a new data set. Which maybe is kind of a strange concept right now, but we'll use this information, we'll use these yreps later to check to make sure that our model generates data that looks similar to the data that we use to actually fit our model. If it doesn't look similar, we have a problem. All right, so let's add some comments in here just to remind our future selves what we did here and what all this stuff means. So generate fake data. All right, well, that's the likelihood. This is the slope parameter. This is the intercept parameter. If you will just note here that these are the priors. This is the likelihood. And we will comment here and say this is the linear predictor, just to remind ourselves. And this is the, maybe this is bird weight here, make that comment. And I'll say bird food abundance. Just so we remind ourselves in the future, it's always good to comment your code, of course, using the / here. All right, well, great. We've got our stand file. What's next? Let's fit the model. All right, so we're going to be using the data that we generated just a moment ago, that we plotted to fit this model. If you didn't work through this with us earlier, there's a script on the GitHub page with all the code up there. So for fitlinearbirdmodel.r, that has the data generating stuff up on it. Go to the morning session, pull this up. All right. So this top here is that same code that we showed before. We're setting the C, we generate the data points, simulate the predictor, came up with some random, well, not random, but arbitrary values for intercept and slope, and then simulate our Y values. So that's just what we did before. So let's go ahead and run all of that. Okay, so we've simulated some data. We're going to load 2 packages here to fit this model. RStan, that is essentially how R interfaces with Stan. You can actually interface with Stan through a couple different programs. And then we're going to load this package MCMC viz. It's a package used to summarize, to visualize, manipulate the output from our models. So kind of a useful thing there. All right, so we've got our data. We need to format these data to provide them to Stan. Stan requires that we provide all this in the form of a list. So if we look back at our model, what are our observed quantities that we need to feed to Stan? Well, it's N, Y, and X. So let's make sure we have all 3 of those things. We have N, yes, as of length 30. Specify bird weight as our Ys. Okay, yep, it's a vector. And food STD here, also a vector of length 30. So putting all these in a list. So data is a list object here, N, Y, and X here. Great. All right, now let's make our function call. But first, before we do that, let's take a quick look and look at the help file for this. So we're going to call the function stan from the rstan package. So the 2 colons here just specifying that the function stan is in the rstan package, just so we're being very explicit here. So let's just run this question mark here. Look at the help file. All right, for the stan function, the first argument is file. It says file is the path to a stan program to use. Well, that's what we just wrote. That is this temp.stan thing. I'm actually going to use a script that I wrote previously. It's already up here, this for linear bird model stan. This is the same thing that we coded up. I just produced it before. I don't want to change the path here. I've already made it. So just what we just produced here, that's the file that we're feeding in. This next argument is data. Look at the help file. Data, a named list, providing the data for the model. So we need it to be in a list. Well, we put it in a list. We did what we needed to do here. We have a couple more arguments. Chains, that is the number of chains, number of different chains that are exploring that probability space when you run your model. So when we looked at those visualizations of the points exploring those probability surfaces, there was just one chain there. There was just one sort of set of points sort of meandering its way down there. But we want to use multiple chains. Pretty common to use 4 chains here, sometimes 3, just to make sure that the output, that all the chains agree with one another and what that posterior distribution looks like. So 4 chains here. Next is iter, that's the number of iterations. That's how long the algorithm should explore this probability space before it stops. We need to tell it how long to explore. It could do that indefinitely. A few thousand iterations is usually sufficient for STAN, but you might need many more iterations than that if you're fitting models using a different program. Like bugs, you might use, tens of thousands or even millions just because of the different ways the algorithms work. For STAN, typically a couple thousand is fine. And then the warm-up here. Warm-up is the portion of the exploration that we actually discard. So in STAN, the algorithm needs to tune itself to make sure that it's sampling that space properly. It actually warps the probability space, that gray surface we looked at, to make it easier to sample. That's some of what it's doing in the warm-up stage. We also want to make sure that the chain has enough time to find the sort of best area of the probability space. So we're just going to discard the first half of the iterations that we run here. It's pretty common to do. We always want to have a warm-up, but half is pretty common here, half of your iterations. And then finally, we have pars. So pars are the parameters, or the quantities really, that we want to track. So we have alpha, beta, and sigma, which are our three parameters here. We want to track all those. If you don't track these things, you're not going to be able to make inference on them in the back end. They won't actually return any results for these things that aren't listed under pars. We're going to track mu, linear predictor, and we're going to track yrep, which is that simulated data we created in the generated quantities block. Okay. We're good to go. We've set all of the necessary arguments. There's some other arguments here that you'll see, but as many functions, we don't need to specify everything. Just go ahead and run this file. Okay, so STAN, it'll take a second to compile the model and then run it. So STAN actually compiles this, and everything is run with a lower level language, not in R, just to speed everything up. This is a very simple model here. So the actual computation time, the time it takes to actually do the fitting, is very quick here. What it's really doing right now is just compiling it. So the compiling might take a few seconds here, but it doesn't really matter how complicated the model is. It's not going to take so long to compile typically. So the runtime here, this is what the output looks like. It's running our 4 chains. Gives you a nice update of where it's at. So if you have a model that's running for a long time, you can check and monitor that progress. So each of these chains only took, fractions of a second to run, which is nice. So real quick here. More complicated models, of course, might take minutes, hours, days, or maybe even weeks in some sort of extreme cases. But anyway, we don't get any errors here or any warnings, which is great. We didn't expect to get any. So yeah, we had a successful sort of model fit here. Before we do anything else, we need to assess if our model is even sort of valid, the model fit is even valid. And there's 4 main diagnostics that we'll look at to sort of check this. If all of these are good, then we're all set to make inference, but we need to check these first. So first off, we have this thing R hat. That is a measure of how well these chains agree about the posterior distribution. Basically, values less than or equal to 1.01 are really what we're looking for here. NF here, that's the number of effective samples from the posterior, essentially how well those chains explore the posterior distribution. You want them to be at least 400 for if you're running 4 chains here, so we'll look for that. Now, R hat and NF are these diagnostics you get for each parameter in your model. So we're going to get one, we're going to get an R hat NF for alpha, we're going to get one for beta, we're going to get one for sigma. If R hat is too large or NF is too small for any parameter, you cannot reliably make inference on your model. So in that sense, it's said that convergence is global in Bayesian models. All parameters have to be well behaved if you're to trust anything about your model, because the value of one parameter is going to be contingent on the values of other parameters. So you want to make sure all of your parameters were sort of behaving properly in your model when we do our assessment here in just a second. For posterior predictive check, we're going to use those yrep values, those sort of generated values, those realizations of the response variables, that we generated from the posterior. So at each iteration of the MCMC, a new data set was generated based on the parameter values at that particular iteration. So if we had 4,000 iterations in our model, we have 4,000 different simulated data sets. And the idea here is to ensure that the simulated data sets look similar to our observed data. And finally, here on the bottom, we have divergences. Divergences are specific to Hamiltonian Monte Carlo. So these divergences basically occur when that posterior density, that gray surface that we were looking at earlier, when it has a very complex topography, like literally complex shape, when it has deep canyons that algorithm isn't able to efficiently explore. That sort of proverbial ball there has trouble efficiently rolling around in those spaces. We don't typically check the divergences. Stan will sort of give us an error or warning when there have been any divergences. So if you don't get any warnings about divergences, then you don't have to worry about them. If you do get some warnings, then there's a few things you can do to address these. I'm not going to say anything more about these today, but know that there's something that you're likely going to encounter if you plan on fitting more complicated models in STAN. And there's some great advice in the STAN documentation for addressing these issues with divergences. So there are certainly common things that you can do to sort of fix your model if you're seeing those divergences. All right. Well, let's assess our model. Let's open our studio back up here. And so the first thing that we're going to do here, we're going to take a look at all these diagnostics. There are a number of different ways we can do this. People have their own workflows. But we're going to use the MCMC trace function here today in the MCMC viz package to go ahead and look at what we call trace plots. All right. Just gave it a couple arguments here telling it to return the R hat and the NF values on these plots. So these are the posterior chains here on the right for our alpha, beta, and sigma parameters. Remember that we specified 4 chains to explore that probability space. Each chain was trying to find the low point and explore that area on the surface. We want to make sure the area that these chains explore is sort of stabilized more or less. And that stabilization looks like a fuzzy caterpillar on these plots. So each chain is a different colored line here. So we have yellow, green, red, and blue, I think, over here. The x-axis is the iteration that it's on. So you can look at this as, think about this as time, right? So as these chains move through the probability space, as we're moving to the right on this plot, we're moving through time actually here. And the value on the y-axis is the value for each of these particular parameters here. So again, fuzziness here, that's all good. These chains look pretty good. We also have the R hat values over here. Well, they're all 1, so they're less than 1.01, which is great. And our NF values, well, they're all well over 400, so that looks good here. Didn't get any warnings about divergences, so we've ticked off all 4 of our different, well, 3 of our 4, I guess now, diagnostics. The one that's left is our posterior predictive check. So the first thing we need to do that posterior predictive check is to extract those Y-rep values from this object here. So from this, fit object. This is our model object, has all of our goodies in it, all the model inference, model results, I should say, in this fit object. We want to extract those values for Y-rep, those data simulated from the posterior. So to do that, we're going to use the MCMC chains function, just specify that we want Y-rep, and it'll give it to us, easy enough. And then what we're going to do is plot a density plot of our observed data. So the data that we simulated in the beginning here, but really the data that we would have gone out and observed in nature. So our observed data here. Now plot the density plot first. And then what we're going to do, we're going to plot a number of different realizations of the simulated data, those Y-reps, and see what it looks like compared to this actual data that we have. We're just going to plot 150 different realizations. We have 4,000 iterations because we ran 2,000 iterations for each chain. So we have 4 chains, it's 8,000 iterations. But we discarded the first half of that, so we're left with 4,000 iterations. So we have 4,000 of these things. We're just going to look at 150 because we just want to get a general sense that that's perfectly fine. So each of these red lines is a realized data set. It's something that we simulated from the posterior distribution. What we're looking for here is generally that the shape of the red lines matches the shape of the black line here. And it does here. You always expect to see some scatter because there's some randomness there. Everything isn't going to fit the data exactly here. There's some residual error, of course. What we're looking for is any sort of biases, strong biases that we might see. If the red lines were humped over here or something like that, well, that would definitely be a problem with your model. For this case, simple linear regression, yeah, of course, we're going to get a decent posterior predictive check. But this is really useful for diagnosing sort of over dispersion, things of that nature. It'll tell you whether you need to maybe add an additional parameter to your model or maybe use a different distribution to model your data. It's really telling you about any systematic biases that your model has. So this looks pretty good. We don't need to be concerned about that. Yeah. So let's summarize this model output now. There's a question in the chat here. What's the difference between warm up and burn in? They're essentially the same thing. The Stan folks call it warm up. Burn in is what the Bugs folks call it. They're technically slightly different, but just know that it's the portion of the posterior that you're just tossing out and not making inference on. Yeah, that's a good question. So let's quickly make a inference on our model here. Let's just summarize it, see what we've got here. We're going to use the MCMC summary function for this. There are other ways of doing this, but this is the method I prefer. I'm the one that wrote the MCMC viz package, so maybe I'm a little bit biased, but I think this is the best way and easiest way to go about this. So using the MCMC summary function for alpha, beta, and sigma, this is the output that it gives us. So let's walk through this. We've rounded it to 2 digits also, just to note. We have our alpha, beta, and sigma parameters here. On the left here, we have our posterior medians. The SD, we have our posterior standard deviation here. And then we have the quantiles here with our R hat and our NFs. So our R hats are all good, they're low. Our NFs are all good, they're high. That's great. So the question in the chat here real quick. NF greater than 400 for 4 chains, then NF greater than 100 for each chain. Thank you. That's exactly right. If you're running 5 chains, you're looking for NF greater than 500, that sort of deal. Yeah, so we're looking good here on all our diagnostics. No issues there. So these are the posterior summaries, basically. We're summarizing our posterior distributions using summary statistics like quantiles. So you typically report the posterior mean or the posterior median and some set of quantiles as a measure of the uncertainty in a publication. And just to note that we'll break here in just a few minutes to give us all a break, but just a few more things to go through here. So just to give you an idea about what's going on here, how to think about these posterior distributions and what these summaries actually mean. Here on this horizontal plane here, this is the joint distribution here for alpha and beta. You can think of each point on the surface as an iteration in that MCMC sampler. So these are sampling different values of alpha and beta here. Yeah, this is the joint distribution. And over here, we have the marginal distributions. So the marginal distributions are what we get when we collapse one of these dimensions. So here we're thinking about sort of a 2-parameter space. Over here, we're just thinking about beta. So it's as if we just ignored the alpha dimension, collapsed all of this down into this density line, into this histogram here, and same here with the alpha. So these are known as marginal distributions, and that's really what we're going to make inference on. When we say that we're taking the posterior mean, we're taking the mean of all of these different values of beta. the quantiles are literally the quantiles of this marginal distribution. So it's the marginal posterior distribution. And these quantiles, these are what we're using to construct credible intervals here. These are going to represent our uncertainty about these parameter estimates. Larger credible intervals are going to mean that you're more uncertain about that parameter's estimate. Narrower credible intervals means that you're more certain about it. So a 95% credible interval would say that there's a 95% probability that the true parameter is within that interval. And it's worth noting that while we often compute the 95% credible intervals, there is absolutely nothing special about that 95% value. That's just a convention. We could just as easily use, 89%. So, 95 is a common choice, but it's only there to assess the uncertainty. Remember, we're estimating these parameter values. We're estimating the effect sizes. We're trying to move away from thresholding. We're trying to move away from p-values in many respects using the Bayesian approach here. All right. All right, so let's just look at one quick look at our results here. Let's plot these here on top of our data. I'm not really going to go through the code here just because it's a little clunky. There's some other stuff here too that we won't have time to go through. So feel free to look through this on your own. We're going to extract the values for the quantiles really for mu, which is our linear predictor, using this MCMC PSTR function. We're going to reorder this the food data just so we can plot it. So these are all of our data points that we generated before, all these grade points, bird weight and bird food. And then we're going to plot our model fit as well as our 95% credible intervals for that fit on this plot. So red line, that's our linear predictor for the model, and the dashed black line, that's the uncertainty about that model fit. So yeah, we fit our first linear model using STAN. We've assessed it, determined that all the diagnostics look good, and then we've plotted our data. All right, well, let's take another 10-minute break here. We'll come back at, let's see, 27 after, let's say, and that'll be the last segment of the morning session. We'll talk about multi-level models and see if we can fit multi-level hierarchical model when we come back. So yeah, I'll see everyone back here in 10 minutes at 27. All right, welcome back. Let's get back to it. A couple questions in the chat here before we continue with hierarchical models. There's a question about the recording, whether this will be recording will be available here. Is TV noted in the chat? Yes, it will. I'll put a link to the recording on the course GitHub page also. So you'll have sort of all the stuff in one place. Might take a couple of days to get that up there, but we'll put that up there. There was also a question regarding whether we need to look at the second order marginals, things like a covariance between these different parameters, or just the, quantiles, the mean and whatnot of the marginal distributions. There might be reasons why you want to understand the covariance between different parameters in those posteriors. But generally, you don't really look at that. If you're having problems with your algorithm sampling, maybe it's not working properly, maybe it's going super slowly, maybe you might need to diagnose some things, but not generally. Another question that just came in, could you explain exactly what's an effective sample? Yeah, so the number of effective samples is essentially how much autocorrelation there is in those Markov chains. So is a chain just sort of wandering around the same area of probability space, or is it effectively sort of exploring a larger area? So is it getting stuck? Is it getting stuck in like a one little area? That is essentially what number of effective samples is. So you want that to be high because you want there to be a decent exploration of that area. That's essentially one way to think about the number of effective samples, but it really comes down to the temporal autocorrelation in the chains. Okay, great. So now that we've covered the basics of Bayes and fit a few models, we can finally talk about hierarchical models that in the title of the workshop here. So what are hierarchical models? Well, as the name implies, it is a model with multiple levels. And I just want to note that these are not inherently Bayesian. Although the Bayesian framework is very well suited for fitting hierarchical models. You can do a lot more with Bayesian analysis. So hierarchical models are known by a few different names. They might be called multi-level models at times, although that makes it sound like some sort of marketing scheme, sort of like hierarchical. And sometimes they're known as random effects or mixed effects models. I have sometimes here because a random effect or mixed effects model is a hierarchical model, but a hierarchical model doesn't have to be a random or mixed effects model. So for example, you could have a measurement error component to your model that would make it hierarchical that doesn't have any sort of random effects or anything of that nature. So that's why I have this sort of sometimes here. So we're going to take a quick look at this link because I think it's a pretty awesome sort of dynamic exploration of hierarchical models. So someone created this, Michael Freeman, I guess. So the way they have this laid out here is that the data are split up into groups here. And if we plot all of our data on a plot, we can see that there's some stratification here. So we have the purple dots, the red dots, the green dots, the orange dots, and the blue dots here, all kind of stratified across this plot. Now we could fit a single intercept, single slope model, kind of what we just did before. Could do that for these data, but we might be missing some structure in the data. So we might then decide that, well, we want to fit a model with varying intercepts, but fit a single slope parameter. Now this visualization uses the term random here, as many people do, random intercepts. I would advocate for simply saying varying intercepts. The term random actually means different things in different fields as far as modeling is concerned. And I think varying intercepts is just a little bit more informative. So I tend to use the terminology varying intercepts rather than random intercepts, but it's really synonymous for the most part, depending on what field you come from. So ultimately, best practices, I think, would dictate that you just write out your model that you're using in your paper so people know what you've done here. I really implore you all to do this. It seems sort of obvious, but I think most of the time people don't. So writing out your model explicitly, I think that's something that's pretty important here. So here what they have is different intercepts for each group here. The example data here is salary. In different academic departments. So we have salary on the y-axis here, years of experience here on the x-axis here. So each of these color groups is a different department. So maybe up here we have computer science or something in purple, and maybe down here in blue we have something like sociology that typically doesn't pay as much for whatever reason. So we might think that, well, some departments just have higher baseline salaries, computer science, than other fields. So maybe we want a varying intercepts model here. but maybe the years of experience, the effect that years of experience has on increases in salary, maybe that's the same across all departments. So this might be a good sort of case for fitting a random intercepts single slope model as they do here. Alternatively, we might say, well, maybe the baseline salary is the same for each of these departments, but the effect of years of experience varies by department. Now, because of what we know about academic salaries, maybe this doesn't make much sense. And, in these cases, you're making less with more experience, but maybe there are some cases where you fit, yes, single intercept, but varying slope. So that would be another option here. Or you might decide that you want both intercepts and slopes to vary. So you know that the baseline varies across departments, but you think that the years of experience, people value years of experience monetarily in different ways across these departments. So maybe you want a varying intercepts, varying slopes model. So this is sort of the most flexible model and something that we sort of, often want to do. We don't know what the baseline and the change in response to our predictor might vary across all these different groups in our data. And I think that these sorts of multi-level models are really a very powerful tool for modeling the structure in your data by accounting for groupings and how things might vary across these groups. I think that this is, excuse me, certainly something that's becoming more important as we continue to use larger and larger and more heterogeneous data sets in our respective scientific fields. We need to be able to accommodate that heterogeneity in our data. And hierarchical models are a great way to do that. All right, so let's jump back to our model that we had before. This was bird weight as a function of bird food. This is the plot that we created before with our model fit, and the uncertainty in our model fit, those 95% credible intervals and the dashed black lines. Here we just fit a single line, single slope, single intercept. So this is the description of that model. So alpha is our intercept, beta is our slope here. But let's now assume that we have maybe multiple species of birds here rather than just one species of birds. Maybe we want to fit a varying intercepts model, but just have a single slope. Maybe the baseline weight varies across species, but the effect of food on weight is the same across species. So some species are just inherently larger or smaller than other ones. So let's let our intercept, our alpha here, vary by group. So instead of alpha here, we're going to have alpha sub j. So alpha j here. J is the index for groups. Maybe we have 5 different species here. So we have 5 groups and 5 different alphas. And we could say here that all of these alphas are drawn from the same distribution, something like this. So each of our 5 alphas is drawn from a normal distribution with mean 50, standard deviation 10. This is the same thing we had before. But there's a better approach than simply doing this. We want to share information across groups. That is, we want to partially pool our data. This partial pooling is what a lot of hierarchical modeling is all about. You're borrowing strength from these different groups. So instead of what we have here, we'll say that our alpha is drawn from a normal distribution with mean mu alpha and standard deviation sigma alpha. So here we've introduced these new parameters. These are known as hyperparameters. These are parameters that govern the shape of a distribution that characterizes some parameters. So we're adding a level here to our model. So this might be considered a hierarchical or multi-level model. And we don't need a prior for alpha anymore. We have something on the right-hand side of alpha here. Alpha is being modeled as a function of stuff, this mean and this standard deviation here. But now we do need priors for our new parameters, our mu alpha and our sigma alpha here. So these need priors. Maybe we give them something similar that we had for alpha before. We just say mu alpha goes as normal with 50, 10. And maybe for our sigma alpha here, maybe we give it a half normal, mean 0, standard deviation 10. Okay, so we have priors for those now. We also need priors for our beta and sigma as we had before. So we can add those on here. This is just the same thing we had before. We're giving our beta a normal prior with mean 0, standard deviation 10, sigma half normal 0, 10 here. All right, so new model. This is our varying intercepts model. We don't have a ton of time left. I was going to see if everyone can sort of work through this on your own, but I think we'll walk through it together for the sake of time and maybe give you some more things to think about on your own after this. These are the files that are associated with this that we'll sort of go through right now. We'll create the stand file based on what we had before, and then we'll go ahead and fit that model as we did before, evaluate it, make inference on it, and plot right there. All right. So let's walk through this model. Let's bring up our studio here. Get my cat off my desk. All right. Second, getting our studio. Find it. There we go. All right, so what we're going to do, let's adapt this previous script. Again, we're just going to sort of use what we've already done to sort of simplify this process, you know. And I will say I do this a lot in my own work. Hey, I'm building a new statistical model. Let's go ahead and use some other script that I've already started with, of course. We don't want to do more work than we have to. So I'm going to take this script and I'm going to save. I'm just going to duplicate this. I'm going to save it as something else here. I'm just going to call this maybe like temp2 here. All right, so this is our sort of starting point to code up this new model here, our varying intercepts model, using the stand file that was just a single intercept and a single slope. All right, so what are we feeding this? What are we feeding this model? Well, we have n here, we have our y's, we have our x's, but we need some sort of group ID, and we need to specify the number of groups that we have for these different bird species. So what we're going to do, say, declare a new variable j, and that's going to be the number of groups. So we'll feed that to our model. It's going to be an integer, just like n. And that's going to tell us how long our vector of alphas is going to be. We also need some index here to specify which observed values are going to be associated with which group here. So again, we're going to specify an integer here. I'm going to call this GID, and this is going to be, comment this, oops. Comment this group ID. So just an index to keep track of which of our observations are associated with which of our species, which of our groups. All right, so I think that's probably good for our data here. Let's go down to our parameters. Before we had alpha declared as a real, beta declared as a real, and sigma declared as a real. Well, sigma and beta look fine here, but alpha is actually now a vector. So let's change this here. It's a vector, and it's going to be of length j. It is going to be equal to the number of groups that we have here. Oh, spelled vector correctly. All right, we also have some additional parameters that we added on here, this mu alpha and sigma alpha. So we're going to have to declare those as well. Let's declare mu alpha as a real. We'll comment this, hyperparameter for alpha, a hypermean. We also have sigma alpha here, so let's declare that as real. So sigma alpha. Mean, call this SD, standard deviation. Well, since this is a standard deviation, just like with sigma, we need to make sure it's bounded on the lower end by 0. So there we go, real for there. Oh, someone in the comments, GID is not a vector of length n. Nice, it actually is of length n. It has to be an integer here. So yes, there will be each Y will have an associated group ID with it. It needs to be a type integer because you're indexing over. So it's technically not a vector. It's annoying, but that's the way that Stan deals with object types. So yes, that needs to be of length n though, not just a single value. So here we have alpha, mu alpha, sigma alpha. We just got our single beta. We've got our single sigma. Looks like we're good there as far as declared parameters. Now let's move down here to the transform parameters block. So we still have our mu. It's still of length n, the same length as y here, same length as x. But this alpha, we have 5 different alphas, or rather j different alphas, one for each species. So what we can do here is just index this by group ID. And I should say that, Stan is vectorized. So I could, say something like this, for I in, length I in 1 to n, we're going to loop through each element of mu and each element of x, etc., and each element of GID. But we don't actually need to do that. We can take advantage of this vectorization. It's a lot quicker. It's a lot more concise programmatically. So yeah, we're going to take advantage of that vectorization. We put the group ID in there to index alpha. So at the first observation, it says, all right, what group does this observation belong to? Is it group 1? Okay, we're going to be referring to the first position in this vector, so alpha 1. We are going to maybe the 31st observation, maybe that's associated with group number 2. So the GID for that particular value is 2. So that's the second alpha. So intercept for the second species. So we're going to put that group ID there to index our alphas. And that's all we need to do there. We're good to go. Okay, so down to our model block. Well, things are a little bit different down in our model block now. Alpha no longer gets this prior. That's not what's going on here. Our alpha is now modeled as a function of, well, it's modeled as normal with mean mu alpha and standard deviation sigma alpha. So let's go ahead and put that in there. It's going to be mu alpha and sigma alpha for alpha there. So that's the hierarchical bit. We have that same first component, that same first level to our model. Our y is distributed normally, mean mu standard deviation sigma. Our beta has a prior, our sigma has a prior, but we still need priors for our mu sigma and our sigma alpha. So let's check what those were. Mu sigma, it's normal 50-10. So put that in there. Mu alpha goes as normal 50-10. And we have sigma alpha goes as normal 0-10. Is that right? 0-10, that's right. And the semicolon. Okay, so let's look at all of our parameters we've declared. Alpha, mu alpha, sigma alpha, beta, sigma. So mu alpha, sigma alpha, beta, and sigma all have priors, and alpha has something on the right-hand side. So it doesn't have a prior, but it is modeled as normal with mean mu alpha and standard deviation sigma alpha. Okay, that looks pretty good to me. So this last section stays the same because this bit is the same. This is just generating random values based on mu and sigma. And this last line is staying the same here. So it looks like we're actually good to go there. There's a question in the chat here. If the alpha GID comes from mu alpha, why do you need to specify alpha as a parameter? Okay, so alpha is an unknown in our model. Remember, we have this worldview of observed and unobserved quantities. Our only observed quantities here are y's, our x's, and our group IDs here, which is sort of, I guess it's observed quantity, your group ID here, and then the lengths here. So these intermediate sort of things here, it could be a latent state, it could be another parameter. This is unobserved, this is unknown. So we're treating that as a parameter in our model. So even if, let's say we've gone out and counted a number of birds, maybe that's our observed count of the birds, but we're interested in making inference on the true number of birds. and we want to put an observation component, an observation level in our model. Well, that true number of birds, that latent state, that's actually a parameter in our model. That's this unobserved quantity. So we would put that in the parameters. So anything that we're estimating, anything that's unknown, that goes in the parameters block. Yeah, that could be a little bit confusing. All right, so we have our stand file here. Let's go ahead and try to simulate some data and fit this model. So I created this script here, this 5var in simulation that simulates some bird weight and food data from 5 different species. The same thing is in the very first section of this 6-fit var int bird model.r. So I won't go through too much detail here, but I'm going to set the seed just as we did before. I'm going to say how many data points we're going to have per group here. I'm going to specify our number of groups, our number of species here. We're just going to say it's 5. Total number of data points is going to be the product of those two things. And we're going to simulate a number of group IDs here. So, just going to repeat each of these numbers, 30 times here. That will feed to Stan, simulate some food data. We're going to make up some parameter values here. I won't walk through this in detail. You can look through it on your own though. And ultimately, we're just going to be left with this, put it into a data frame so it's easy to visualize. This sort of 3 different columns. We have our bird weight here in the first column. Those are our response variables, our food, our X's here, just as we had before. But now we have this GID, this group ID. So this tells you for each observation, for each row, what group that belongs to. So these first ones, they all belong to group 1, species 1. These all for species 2, and so forth. All right, so we've got our data here. We're going to load these packages, rstan and MCMC viz, just as before. I'm also going to load the tidyverse package just to do a little bit of processing for the plotting at the end. It just makes it a little bit easier. We're going to feed our data into a list here. Let's make sure we have all of these things in our model. I'm actually going to open up this other stand file. This is what we just created, but this is something I created earlier. I just want to make sure this works properly. It's the same thing though. In our data block, we created object n, j, x, y, and gid. nj, x, y, and gid. Okay, that's everything we need there. Those are all our observed quantities that we're feeding our model. We then are going to call our stand function from the rstand package. It's going to actually fit the model. This is the same thing we had before, except we're then careful to add these additional parameters that we added to our model. So we need to track this mu alpha and sigma alpha that we added to our model because we have this varying intercepts now. So go ahead and fit that. It'll take a few minutes just to compile the model as it did before, and then it'll go ahead and run through that pretty quickly. We can then take a look at that output. Again, we'll look at those trace plots there. We'll be looking for fuzzy caterpillars. That's what we're hoping for, that the chains have sort of stabilized in the posterior space. Okay, no errors, no warnings. That's a good sign here. Still ran very quickly, less than a second here. Again, these are pretty simple models, but it's good to see some quick models here. We like that. Okay, let's run through the trace plots here very quickly. So we have 5 different alpha parameters because we made up the data. So there are 5 different groups here. Those all look pretty fuzzy to me. Let's look at our other parameters. Mu alpha looking good, sigma alpha, beta, sigma. Oh, okay, those all look great. Our R hats and our NS look great for each of these parameters. Just to note, these are the marginal distributions of each of these parameters. So this is summarizing these trace plots right here, essentially. So the mass of this posterior distribution is centered around 3.2. And you can see that these chains have sort of stabilized on 3.2. So they're sort of wandering around the probability space just around 3.2. And if you're asking yourself, well, I still haven't got a good idea of why we ignore denominator in Bayes' rule, well, this is really the reason why. It doesn't really matter what the probability is at any point in this iteration. It just matters how much time these chains spent in each of these areas of the probability space. So this is just a density of the time, basically, that these chains spent in these places. So if anyone's still wondering about that, that's sort of a good way to think about it, I think. Let's extract our simulated data, our Y-reps here. Go ahead and plot that for our posterior predictive check. We ticked off our R-hat, we ticked off our NF. We ticked off our divergences. We didn't get any warning about divergences. So the last thing is to check our posterior predictive check. Well, our red lines look pretty good. They look like they're, matching the black line pretty well here. There's a little bit of variation that we're not picking up, but, we expect that sort of thing. There's no, big bias here in our model. So, we're free to sort of, move on to interpreting our model fit. The question in the chat here, does Stan accept data as a data frame or just as a list? I believe it has to be a list. I don't know. I guess I never tried it as a, it would have to be a list because different elements can be of different dimensions. So n would be, of length 1. In this case, our y's would be of length 30. So yeah, you'd want to use a list. Maybe you could use a data frame if they're all the same length. I'm not actually sure about that. So we can summarize our data just as we did before. Same thing, posterior mean, standard deviation, the quantiles. These are our diagnostics here, our R-hat and our NF. Everything looks good. We already saw that on our trace plots though. One thing that you might want to do is to plot your data as caterpillar plots. So this is a common thing that folks often do. The parameter estimate here is on the x-axis. Each of these lines is associated with a different alpha. This one has the new alpha. The dots here represent the posterior median, and the length of the lines represent the uncertainty about these parameters. So the thin lines are the 95% credible intervals here, and the thick lines are the 50% credible intervals. So that's just a common way of visualizing your uncertainty in your parameter estimates, particularly for a hierarchical model. Real quick here, we'll sort of plot our model fit. We'll go through the code here. But feel free to look through this on the script on your own. So here we're plotting each species, each group as a different color. We have varying intercepts for each group, as we set our model up as, and we have the same slope here. So our red species down here, whatever group that is, that intercept is around 32 or something. So that would correspond to alpha 1 here. If we go back to our posterior summary here, so we can make all these match up, but yeah, and these all have a slope about 3.12. So we're looking good. And well, that is our varying intercepts model. So hopefully that wasn't too complicated. It's a fairly simple model. I hope this makes it clear how we can build upon the structure and add more levels. In fact, we might model parameters as a function of other variables. So we could have predictors on 2 different levels instead of 1 level. And I think the Bayesian framework really opens up a lot of opportunities to sort of be flexible, sort of as we sort of opened up with, account for these measurement errors, account for latent states to forecast, et cetera. Yeah, so we have a couple of questions here. Can you explain what it means that there are 2 peaks in the posterior density plot again? Yeah, 2 peaks in the posterior density plot. That would mean that you have a bimodal distribution. You're sort of exploring 2 different areas of the probability space that might signal that you have some sampling problems in like the computation of your model, or it might signal that your data have a multimodal distribution. It really depends on your problems there. All right, let's get rid of this for a second. Just a quick few things before everyone goes here. If you continue to explore Stan, I greatly encourage you to look at the documentation. It is really excellent. There's a number of different user manuals. There's a pretty active Stan user community. Somebody asked, is there a Python equivalent package to RStan? There is. It's called PyStan. So you can use Stan. You'll build those same Stan files, but just interface with Stan using a different interface, either the command line, Bash, or something like Python. I think there's even a Julia version if anyone uses Julia. Yeah, so a lot of good stuff in the documentation, so check it out. There are also some packages that sort of provide an abstraction to Stan called BRMS and RStan ARM. These allow you to use sort of like an LMER-like syntax. I don't really recommend using these tools to start because you want to write out the full model code. You're going to be writing out these models on paper anyway, so doing it in full Stan isn't too tricky, I think, for the most part. But these are there if you want a little bit less flexibility, but maybe a little bit more usability, so you can check those out. For general Bayesian resources, I highly recommend all three of these books: Hobson, Hooton, McElreath, and Gelman et al. These are all excellent books. I would say Statistical Rethinking is the most accessible of these, but they're all sort of taking a different perspective on Bayesian stats. So we've gone through quite a bit of material over the last 3 hours, probably about a half a semester's worth. My hope here is that whatever level you're coming in with, that you're taking something away here, and you'll be able to come back to these slides, sort of understand some of these concepts you didn't fully grasp. Hopefully you now have a better grasp of the material. Right before we break for lunch, I'd just like to say thank you for attending this part one. If everyone could fill out this anonymous survey, I think that would be really helpful just to get a better sense of what went well, what went not well here. Your feedback would be very much appreciated. Again, totally anonymous. It takes about 2 minutes. Hope to see everyone back here at 1 for the spatial analysis portion, led by Sudipto Banerjee. It's going to be some pretty cool stuff there, I think. I'm going to take a 1 hour break for lunch, 12 to 1 here, and we'll come back at this same link. I'll hang around on Zoom for a couple minutes. And again, I'll post these slides up on GitHub and put a link to the presentation here within the next couple of days. So take a look for that. Yeah, and thank you all.
Thank you, Casey. And we will be back at 1 o'clock.
Just go ahead and answer a couple of these questions too. Let me see if I missed any. Let's see. Same fit statistics. Yeah, exactly. Same sort of summaries for the marginals, for the marginal distributions.
We can start, I think. Let me introduce you. So welcome back. It's been a really good session, the previous session, and I look forward for the exciting session that's coming up. So our next presenter is Professor Sudipto Banerjee. He is a professor and chair of the Department of Biostatistics in the School of Public Health. He is current president of the International Society for Bayesian Analysis. Professor Banerjee earned his PhD from University of Connecticut in 2000, and he has worked at University of Minnesota before joining the UCLA as a professor and chair of biostatistics. Professor Banerjee received many awards, including the Abdel El-Sharawi Award from the International Environmetric Society, the Montemer Spiegelman Award from the American Public Health Association, and the George W. Senedekor Award from the Committee of Presidents of Statistical Societies. He has many more honors, including Distinguished Achievement Medal and Outstanding Statistical Application Award from the American Statistical Association. There is a long list of research articles, including popular books he has published. His current research interests are spatial statistics and geographic information systems, Bayesian statistics and hierarchical modeling, and scalable Gaussian process model systems. uh, process models for big data analysis. We are lucky to have him to talk about the hierarchical Bayesian modeling for spatial data science. So please join me in welcoming Professor Banerjee. Professor Banerjee.
Thank you. Thank you so much. And welcome. I'm sure this is not going to be as good as Casey's performance in the morning, but I will try to give you guys an overview of spatial data analysis and some of the resources that we have to analyze spatially referenced data. As TV very generously introduced me, I have made a living out of doing spatial data analysis. And I don't know where you guys mostly are, but if you are mostly in and around the UCLA campus, and if you are in doing research and analysis with spatial data, feel free to drop by my office. We are, as Stevie mentioned, in campus very much in the CHS building, which is basically opposite the parking garage number 9. And we can, I will be happy to talk to you more about that. I also teach a course in my department, but I don't offer it every year because I have to teach some of the core courses as well, which is on spatial data analysis. And it's, I think, called biostatistical spatial data, or rather spatial data analysis for biostatistics. I forget the exact name, but you can almost call it spatial data science. And it's not, previously it used to be taught at a very technical level. So mostly folks with a background in theoretical statistics or mathematics or computer science with a fair bit of math training would be more appreciative of that. But over the last 2 editions, particularly since COVID broke out, I've been teaching it in Zoom, and the course has really become much more of a computational nature. So I actually literally teach through demos and so on, almost the entire course, and bringing in slides here and there. There will be some equations here and there, but mostly at a conceptual level and a computational level. So no matter what your academic preparation or your academic training is in, it's if you are interested in GIS data and particularly doing Bayesian modeling for GIS data, you may find that useful. The slides I'm going to present today, they're pretty much all hosted. Casey has very generously placed a link to that. You can just download the zip folder containing the files and computer programs. I don't even think I will be able to get to all of them because usually, as I said, this topic requires at least a 1-quarter course, preferably a 2-quarter course, even leaving out the mathematics of it. And you know what, and there Casey actually very efficiently put that on the chat box too. Thank you, Casey, for doing that. But what I'll try to do is give you an overview of what it is like to be looking at spatial data using geographic information in data analysis or data science, and then we'll take it from there, and I'll try to give you a couple of demos in R as well. Okay, so first we'll start with what is spatial data, and the way we look at it is any data with some geographical information. Now, I will say that when we look in the world of statistics, we usually talk about geographic data through GIS, so that means it's over the earth. Either it's a region referenced through maybe counties or census tracts, or it could be just GPS coordinates, whatever it is. But in terms of the modeling, and particularly in the sciences, spatial orientation is a little bit broader, but it's very connected to this. It could also mean something that is not in geographic space. Like, for example, nowadays, you have social network models and so on. And there you have these networks where you are connected to someone. Basically, in the language of spatial data analysis, we say you are someone's neighbor, but that does not mean you're a neighbor in geographic space, but that could mean that you are connected to this person through your Facebook or some other social media platform. And perhaps you've had massive interaction, or a certain volume of text has being exchanged between you and this individual. So it doesn't necessarily have to be geographic. However, the terminology does come from geography. The history is geography, and it's about essentially being connected or being a neighbor of or being proximate to someone in some kind of space, which is what modern spatial data is all about. Oh, let me put this in presentation mode. So however, in the sciences, and particularly here, you have within the UCLA campus, you have the Department of Statistics, you have the Department of Biostatistics, you have the Institute of the Environment and Sustainability. In all these three institutes, I actually have joint appointments. And you also have, of course, IDRE, which is bringing in a lot of research together, and then computer science, and then the natural sciences, the physical sciences, where spatial data actually plays a very important role. So there are many common sources of spatial data, and they include climate science, forestry, ecology, environmental health, and disease epidemiology, which are very much within the School of Public Health, real estate marketing in business, where you actually look at units, like maybe in real estate marketing, all of you probably are familiar with Zillow or some such mechanism where you put in your home address and it actually gives you an estimate of how much your house is going to sell for if it were in the market today, right? So how does it do that? Well, it actually uses a kind of a spatial algorithm, which basically takes in information from the nearby homes, averages it, and basically gives you the estimate. Now, there are better ways. How do you improve this? What's the right way to do statistical modeling of this? So if you want to do that as an amateur, you can do pretty much any algorithm. But if you're in the business of either doing research where you're trying to publish in a scientific journal using spatial data analysis, well, then you will need the statistician or statistical rigor to try to make sure that you have the right framework for your conclusions. Or even if you're in the private sector and you're working for a company, you might want to look at more formal methods or more efficient methods that will improve the inference or the conclusions from what you would get by doing something rather ad hoc. And so typically these sorts of GIS data or databases, they have many important predictors and response variables, and they're often presented as maps. And as I said, sometimes, broadly speaking, if you broadly interpret spatial data, then it could refer to situations where it's not necessarily geographic space. So for example, if you're in neuroimaging and you're looking at data for each voxel in the brain, well, the voxels are situated inside an organ, and you basically want to understand how to model proximate behavior across voxels in the brain. If you're doing genetics and you're trying to look at some sort of chromosomal analysis, you might want to be actually looking at positions along a chromosome and trying to see make sense out of your molecular level or micro level data, as the case may be. However, for the purpose of this talk, most of the examples that I have, we are going to be looking at this point referenced spatial data for the most part, although I will talk a little bit about aerial data and mapping in R as well. So each observation is associated with a location in point reference spatial data, and this is called point. Right. By the way, if you're wondering what slide I'm looking at, it's the file called Intro to Geostatistics, Introduction to Geostatistics.pdf in Casey's GitHub folder. So the data represents a sample from a continuous spatial domain, and it's often referred to as geocoded or geostatistical data. So here, for example, is an example of how you display such data. And everything that I do here, I use only the R statistical computing environment. We have authored packages, me, my students, and collaborators in different stages of my life. I obviously don't program as much as I used to when I had a lot more hair on my head. But I'm still very much involved in algorithm design and statistical software development. And while beneath the layer, you may have some C++ algorithms or C code to actually do efficient computations, particularly for big data, the R packages that we use or that we have ourselves produced are user-friendly to the extent that if you know R and you read the documentation, you will be able to run them from within R. You don't need a working knowledge of C, C++, or Java or anything else. Okay. So this is a very typical way of plotting this. And what we do is these are the points where the data have come from. If you're good with your geography, you will be immediately able to identify this region. This is Western Europe. Here's the United Kingdom, but we haven't taken any data from the United Kingdom. We've taken particularly data from Western continental United States. So you have Portugal, Spain, France, Germany, Italy, and some of the lower Scandinavian countries. And these are pollutant levels. So this is PM 2.5. One easy way to plot this is to, first of all, project the region onto a map so that we are in planar coordinates, easting, northing. You could do geographic coordinates. You could have a different type of projection. That's fine. But in terms of, looking at a region in the Earth, which is fairly localized, such as this, where our concentration is in industrial Europe, and trying to understand where levels of pollutants are very high, it makes life a lot easier if you just project it into sort of a UTM style coordinate system or something like that. And here, you will have you have a little graduation of a sidebar, which goes from deep blue to deep red, with deep red indicating high levels and deep blue indicating very low levels of pollutants. And essentially, you can see, even without doing anything else, you can see the regions where you are actually looking at data where you have higher number of pollutants, so higher levels of pollutants. So for example, the key point here is that this is unfiltered, unsmoothed, raw data. All right, this is exactly what has been provided to us from the governments of these countries and which are part of the European Union. And they're basically telling us, look, we need to analyze this data set on a daily basis. Can you help us? And so what the first thing we do is we actually plot the data. And there are many ways to plot the data without doing any type of filtering or smoothing. Just look at the locations and then basically try to see where the levels are high or not. So we'll see a little bit more of these different sorts of things in R when I do a couple of computer demos. But there are some conceptual levels you need to understand, whether you're doing methodological data science or whether you're doing applied data science. It doesn't matter. That only affects in terms of how you want to present, or if you're doing a methodological paper that you want to publish in a theoretical journal, that's a different matter altogether. But even if you want to analyze data, all right, no matter whether you are a theoretical statistician or whether you're a complete user-based applied data scientist, it's totally fine. But you still need this conception as to what type of data, what type of referencing are we going to do? Because the one thing that spatial statistics has taught us over the years is that variability matters, and variability depends upon what type of data we are looking at. So if you're looking at point level modeling, so for example, the first thing obviously is, what is the spatial referencing for your data, right? And if the spatial referencing is with respect to coordinates, you see I put this in red here, that means it's either latitude, longitude, or easting, northing, or some kind of coordinates, a point, then we are referring to that as point level modeling. All right, and this point reference data is collected at locations. Remember, these are usually fixed locations, and you have observed some value of a variable of interest, of scientific interest or of business interest, whatever it is, at that location. So in this previous example, the values that we plotted here are PM 2.5 levels, of course, scaled up to a certain level, but it could be the particulate matter 2.5, maybe measured in micrograms per cubic meter or something like that in terms of the density. There are many different metrics. Whatever be the metric, the important thing is you have a value. And there is one other important concept here, which is the concept of a process. All right. Now, you will hear this in a lot of presentations and talks, whether you're talking to ecologists or whether you're talking to econometricians or whether you're talking to climate scientists, they all talk about processes. So what is a process, right? A process in data science essentially means a data generating mechanism. All right. So what that means is that you have a domain D, and then within this domain, you have different point reference locations. And at each location, you actually have a measurement. All right. That's what it is. Now, you would not have observed the entire data, possible data, because that would mean a smooth, complete surface here over the entire domain right here. In other words, because this is a pollutant level, and conceptually the pollutant level exists at all possible sites. It exists everywhere, not just where I have observed it. And this brings us to a very important aspect of spatial data science, which is interpolation. So yes, I have observed this only over a small set of locations, but folks want to know about these levels pretty much everywhere over this region. So how efficiently can I actually take the data that I have been provided at finite number of points here, and then completely in continuum predict or interpolate the pollutant surface over this entire domain, so that anywhere, whether this is a residential area or anywhere, if somebody wants to know what the expected value of the pollutant should be, then we can provide them that. With, in statistics, and in particular in Bayesian statistics, as we try to emphasize, with uncertainty quantification. So, okay, it's not just the prediction, but it's the entire predictive distribution. It's the entire distribution of the variable that you are looking at, that you want to understand, that you want to predict, conditional on all the data. And so the statistical objectives include inference about the process, and predicting at new locations. All right. And what is remarkable is that we can learn, literally probabilistically learn about the entire YS surface. And I'll talk about how that is done in a few minutes. But I do want to make one point clear. Many of you may be coming in from a machine learning perspective to this. And sometimes I get this question, what is the difference between machine learning and statistics? And to be honest, if you're a Bayesian statistician or a probabilistic machine learner, then you are pretty much doing the same thing. The algorithms that you choose to implement may be a little different, but overall your line of thinking in terms of analysis of a data set is very similar. However, both statistics and machine learning are not restricted to probability and Bayesian statistics. Statistics obviously relies a lot on probability, very advanced probability, in fact, where we try to understand laws of large numbers and so on. But importantly, there is outside of Bayesian statistics, there's classical statistics, which in fact most of you have some familiarity with, I would imagine, which is usually taught in the elementary stat classes, where you have a null hypothesis and so on. Bayesian statistics is quite different, and I'll talk more about Bayesian statistics, of course, in the next segment. Similarly, probabilistic machine learning is one part of machine learning, a very important part, where uncertainty quantification and scientific machine learning comes in, because it provides you some measures of uncertainty with regard to what you are trying to learn. But probabilistic machine learning and Bayesian statistics are very similar. Pick up a book like maybe there's a book called Machine Learning, a Probabilistic Perspective, or there's another couple of books on pattern recognition and machine learning by Chris Bishop and Kevin Murphy. These guys, you can just Google them. You will see that almost the entire book is doing Bayesian statistics, of course, with different features, but the overall paradigm is very similar. So just keep that in mind. And if you are familiar with probabilistic machine learning, some of the things I will say here and in the subsequent lectures will be familiar, except that the jargon or the language or the words that I will use may be a little different. So the key here is structured dependence. And what do we mean by that? All right. So let's take a very simple example. All right. So here you see I have a data observed at n locations. They're somewhere hidden in here, the n locations. All right. At each SI, we observe the response, Y of SI, SI being one of the locations here in this finite set. And then we also observe a covariate, all right? Covariance, P by 1. Now, let us say, take a very simple example where we have one response and one covariate. So one of the things that you can do as a descriptive analysis, even without doing really sophisticated spatial statistics, is to just create an interpolatory plot over the region of interest. So that means you basically look at the points and then you simply look at bilinear interpolation or some kind of Kriging interpolation, which basically is going to give you a surface such as this. So it will try, based upon the data, to try to say, okay, here's what it looks like. But there is no uncertainty quantification. We don't really call this Bayesian inference. We don't call this inference. We call this exploratory data analysis, or EDA. That's what this is all about, okay? So you actually can plot this, and I will show you, how to do this in R. And then similarly here, you have the covariate, right? Okay, now look at this pattern here. If you notice, you have a nice, interesting segment of blue here. Blue is low. See, these are pretty much the same graduation in color from deep blue to deep red. Blue is means low and red means high, whatever the value is. This is an artificial data set, all right? So when the covariate is high along the spatial segment here, you basically see that the response is low. And when the covariate value is low right here, you basically see the pattern that the response is high. And similarly, again, when it is high here, it becomes low here. So this is very typical of where you have a negative association, as we like to call it, between Y and X. A classic example, I mean, you can think of many examples. The most obvious one, particularly from climate science, would be elevation and temperature. If this is elevation and this is temperature, you would probably see something very similar. If elevation is high, temperature tends to be low. If elevation is low, temperature tends to be higher. So that's one example. Now, what's so special about spatial? We often like to say this because spatial is an investment. After all, is it worth looking at spatial dependence? That's an interesting question, isn't it? So in many cases, you don't really need that, right? I mean, for example, yes, we do live in a world of space and time, right? Of course, we know that. Albert Einstein proved that, basically. But for statisticians, spatial statistics will make sense only if the data that we are exploring, or that we want to model, or that we want to understand, if that data has spatial structure and there is spatial dependence. Because if not, then we could just do ordinary least squares. And this is everywhere in data science, right? I mean, everybody knows nowadays how to do this. It's remarkable that it took statisticians 20 years to basically figure out the ins and outs of this. In fact, as a graduate student almost 30 years ago, I was once listening to a popular lecture by a visiting physicist from a very famous physicist from a different university where I was. And the audience was asked the question, what is the biggest contribution of statistics to science? And there were different answers that were floating around in that room, including chi-square tests, goodness of fit tests, and this and that. But the speaker said that he thought that it was least squares because statistics helped us understand least squares. Now, remember, Newton actually used least squares in his research to try to understand gravitation and so on, and even in his research in astronomy. And perhaps it even preceded him, I don't know. But to understand exactly the properties of least squares and distribution theory associated with least squares, that's what linear regression is. So all you do is you take data, put that in a vector, take a matrix of predictors or explanatory variables or covariates, whatever term you want to use. Some people just like to say dependent variable. This is the dependent variable because it depends on X, and X are the independent variables. So that's fine. And then you basically solve a least squares equation, which is like this. You never really quite take the inverse. You actually solve the system, X transpose X beta equal to X transpose Y. These are known as the normal equations. And then if you are doing statistics, you will assume that this error comes from normal distributions, and you will basically try to understand confidence intervals or uncertainty quantification or standard errors of your estimates. And then using those, you can then do predictions and look at predictive uncertainty and so on and so forth. So this is one slide just to say that, well, you would just assume IID and then you would do this. Okay, so what happens then? Well, you see, let's suppose I do this for this data set, okay? And then I take the residuals and then I plot them here. Well, what we see is that these residuals, when I plot the residuals on an image plot or through an image plot here, there are some apparent clusters, but there's a lot of randomness. And clearly there may be some latent spatial patterns which may or may not exist, right? So here, in this particular case, if you look at this, the right way to interpret this picture is that this is pretty random. I don't quite see a very prominent spatial pattern left. And why is that? Because if you just go back, pretty much the variability here, as you see, right, is pretty much explained by this covariate. So in terms of the spatial pattern, the angle at which this is coming down, where this is high, where this is low, is almost very completely explained by this predictor. So therefore, when I do a spatial linear regression with a predictor that is spatially informative, and provides enough information for the data to move forward, you don't need anything else, right? So that's why you get a random surface of residuals. And you might say, well, but Professor, what about these little clusters here and here and here? Yes, there will be some noise. But remember, look at the scale here. It's going from -0.2 to hardly 0.2. It's not large. And it also is scattered all over. And so this is a classic example of random residuals where there really is not as much of a pronounced spatial structure. You can still fit a spatial model, but it's not clear it will really explain anything very prominent, and its spatial models are more expensive to run, computationally speaking, I mean. And so perhaps it's not worth the trouble. So if your boss gives you something like this and you see a pronounced covariate that's explaining away the spatial variation, go for it and don't need to get any more fancy. But then you have to ask a question, especially us in statistics or data science, you will ask, well, but does a non-spatial regression model always suffice, right? And so here, let's take an example, another example, okay? So here you have a data that consists of a census of all trees in a 10-hectare stand in the state of Oregon. It's called the Western Experimental Forestry Data. It's actually available in our package called SP Bayes. And one of my former students, Andrew Finley, who will soon be collaborating with Casey, I believe. Andrew actually is a forester, and he's spent his entire career actually doing data science on forestry, and has a lot of such data. So here, let's take a look at this. This is diameter at breast height for the tree, okay, for different species. As you can see, there are different species. These are color-coded. There are 4 different species here, right? And now, if you notice, you actually, let's say you regress just on the species, treating these as categorical variables. You regress the information on species or the type of species on this continuous variable, okay? So this is almost like an ANOVA. Remember, in regression, when you have continuous measurements and categorical predictors, that's basically what is ANOVA. So sometimes people will talk about ANOVA by species. So they want to understand the variability by species on the response, how each species is in fact influencing the mean level of DBH. And diameter of breast height is basically the diameter of the tree at, I think, around 4 meters from the ground or something like that. And then, you have these residuals, and the residuals are like dark blue to dark red. Again, but you see, you have a lot more spatial clustering, smoothing going on here in the residuals here. This is not, mind you, any statistical smoothing. It's just the residuals plotted on at a high resolution as an interpolated map through here. And this interpolation is purely deterministic just for exploratory data analysis. So now you tend to see local spatial patterns in the residual plot. Right here you have spots, right here you have spots. There's no randomness, there's lots of smooth spatial variation by species. So a simple regression on species seems not to be sufficient. So in other words, what does this simple exploration help us do? It tells us that we are able to look at a residual from an ordinary least squares model, a simple linear regression model, and we can say whether there is variation, whether there is patterns, and so on. Is it worth looking for other covariates that might explain some of the variation that we see in the map on the left-hand side? All right, so that's clearly an example. Besides eyeballing these residual surfaces, how do we do more formal exploratory data analysis to identify spatial pattern? So if you're working in a GIS company, or if you're working with geologists or geographers, sometimes they will want to understand a bit more quantitative analysis, a bit more objective analysis than simply everyone looking at residual plots and bringing their own interpretation to the table. Some things we can definitely agree on. Like, obviously, there is a lot of spatial smoothing. I don't think anybody looking at this residual plot will say that there is no spatial structure here. But, like, why is there spatial structure, or to what extent is the spatial structure pronounced? Those things, could be subjective and vary from person to person. So how do we do more formal EDA to identify spatial pattern? And here we come to the literature in geography, and we come to the first law of geography, which basically says everything is related to everything else, but near things are more related than distant things. And this is a beautiful quote by Waldo Tobler, all right, who was one of the finest geographers of his time. So when statisticians look at these sorts of sentences or philosophies, if you will, what we try to do is try to understand and translate that into the language of mathematics and try to see what does that really mean. Okay, so everything is related to everything else, but near things are more related than distant things. Well, so what does that mean? That basically means that in general, if I look at a value of y of s plus h, and then I look at my y of s, all right, s plus h being a location that is displaced from another location, depending upon the distance or separation between these 2 points, the distance in the values between these 2 points will vary. So in other words, if h is small and therefore near things are more related than distant things, then I would expect y of s plus h minus ys to be very small, and therefore this value would be small. For those of you who like to think in terms of calculus, well, if h goes to 0, and if this is indeed a continuous process, then frankly speaking, these will coincide, in fact. But on the other hand, as the separation starts increasing, right, this value starts increasing as well, and that actually implies spatial correlation. So can this be formalized to identify spatial pattern? All right. How am I going to take data and just do some data science, exploratory data analysis, not full-fledged statistical modeling, to try to understand what happens? So one way to approach this is to create what are known as empirical semi-variograms. What do we do with these? So we take intervals, right? So the first interval from 0 to M1, the second interval from M1 to M2, and so forth, up to the kth interval, right? Mk minus 1 up to Mk. And then we represent each interval by its midpoint, okay? And we create these sort of bins. As you can see, these are all points that lie within this interval. And what we do is then we basically average this quantity over the bins. That's all we do. So in other words, that's the formal way to write this. This is the sum of this value, the distance between the values, or the squared difference between the values of all pairs of points that lie in this bin, which you could also call a neighbor set, meaning you will treat points within this neighbor set to be like neighbors. That's why sometimes they use the label n and call this that. And then divide by the total number of points in that neighbor set. And by historical tradition, you put in a 2 here to make this semi. If this 2 wasn't here, then this would be what is called a variogram. So you could plot a variogram, and then you could see if there is, if for spatial data, this gamma of tk is expected to roughly increase with tk. If that happens, then clearly there is a well-behaved spatial correlation. Because remember, when this value increases, that is what implies spatial correlation. So this leads us to a very nice plot. And here you see the data when the, for example, if you look at the first example, all right, that I showed you in that simulated setting where the covariate is explaining everything well. If I look at the semi-variogram for the y, that means just do this with y, well, you do clearly see spatial patterns, isn't it? Like here is a spatial pattern, so you will expect spatial association strong here, although this is low values and this is high values, but they're still, in terms of correlation, they could still be pretty correlated. So therefore, you get to see the semi-variogram go up and rise. And if you wanted to fit this, it's possible you will fit a empirical or theoretical variogram to this as a best fit, and you can talk about that. Then when the residuals come in, which are the residuals for, not from this example, but right here, my goodness, you don't see much, because there's really nothing. There's no increase. So basically, this is a way for you to say that, what, no need to look at spatial modeling any further. We will just look at the predictors that are after all spatially indexed, and those predictors themselves are going to actually be enough to explain spatial variation. So that's what happens. The residuals don't really display much spatial variation. But now take a look at the second example where you have diameter of breast height and residuals. Now you see you have DBH also gradually increasing, but after even explaining for the species, doing an ANOVA on the species, the residuals still keep increasing. So that means here, clearly there seems to be unexplained spatial correlation, all right? So that's basically what is going on. So now we come to modeling, and what we are going to try to do is we are going to say, well, let us in fact take the basics and let us in fact insert a spatial process. So that is basically going to help us explain the generation of the data that has manifested itself to us, all right? What is the underlying process? And so what we do is we say, all right, fine. So we will have the regression equation, but we will insert an unknown function of S. And this is, by the way, where statistics actually did have an important role to play. Because if you look at the traditional applied math or the subject matter literature, either in geography or in geology, folks used to interpolate this using some kind of like a bilinear function, which is essentially a product of a linear function or a linear interpolator in each axis, or people would look at bivariate splines and something like that. For what statisticians did to the scientific community is through their own calculations, explorations, they figured that if we actually do not specify what we call a parametric form for WS, so that means, no, it's not like a linear or quadratic or anything like that. We let it be unspecified, but let a random stochastic process generate its behavior, then we get much better interpretation of the variability of YS, and in terms of goodness of fit, as well as in terms of the ability of the model to predict or interpolate, which you can always test using holding out some values from your data set and then trying to predict at those locations and see how well they do in terms of the values of the variable at those locations. So essentially what we want to do is we want to model a surface, right? And the question is, how do we do this? So now we come to a slightly technical side of this, but it's really not that difficult as long as you understand the concept, which is what I'm trying to drive here. So you basically, what you have is you have a W of S, which is an unknown function, okay? Now, as Bayesians, if you have an unknown parameter, for example, you put a prior distribution on it, right? And Casey, I'm sure, mentioned this in the morning. But when you actually have a random surface or a random function that you do not know how it is and how it will behave, right? Then you actually need what in probability is known as a stochastic process to model that unknown function. And the Gaussian process is a particularly popular machine learning tool, or a stochastic process is widely deployed in machine learning to try to model unknown functions that are real valued without a lot of information. They're extremely adaptable. They give good predictive performances, and they're also kind of well understood. So it's a valid stochastic process. And what a stochastic process does is it models not just points at a finite set of locations, but an entire set. But once you've specified this to be a Gaussian process, the good news is that no matter what n locations you look at in that domain, the joint distribution here will always follow a multivariate Gaussian distribution. And a GP is completely characterized by its mean function and then also a covariance function C. So there is a lot of advantages here. If, for example, your W were observed, all right, that means this is the data, let's say, and you directly want to model it, well, then you could do likelihood-based inference directly from here because this guy basically follows normal MC. But of course, usually the W is latent. And in Bayes, what we do, and I'll show this to you in a later segment today, is that we're going to put a prior distribution on this, which is what we have here, right? A distribution. And then here's a mean function. And then this guy is a very interesting one. It's called a covariance function. So it actually gives you the covariance between WS at the value of W at SI and the value of W at SJ. This function you specify. And there is obviously a lot of rich theory about covariance functions, but for the purposes of this course and for most practical data analysis, imagine you are already given a bag of covariance functions. You just choose your favorite model. And then, so there is this question though about that, you cannot imagine any covariance function. You cannot imagine any function and say, oh, that will be what I'll use to model a covariance function, because this function, as you notice, determines this matrix, the covariance matrix. And as we all know, a covariance matrix needs to be positive definite so that the likelihood can be computed correctly. Okay, based upon this covariance function, you're going to have different assumptions for your latent process. So for example, if the separation between the two locations depends only on the if the function depends only on the separation between its two arguments, then we say that it's stationary. If it depends only on the distance between the 2 points, then we say it's isotropic. And then finally, anisotropy is another method where we completely abandon this assumption, and essentially we say, abandon the assumption of isotropy, but we say that it will still be stationary, but it's not isotropic. So isotropic models are popular because of their simplicity, their interpretability, and because a number of relatively simple parametric forms basically can serve as candidates for C. So here is a brief table of parametric forms for covariance functions. So as you can see, they usually start somewhere, and then they tend to decay depending upon the value of the phi. And then basically after a while, they basically they basically go to a distance, like for example, not after a while, when they're close to zero, they basically have just a value here. So what is this value tau squared plus sigma squared? If you go back to a picture of the variogram, it's basically this value right here where this variogram hits the ceiling. And what you have is the tau squared, which is called the measurement error variance, which seems to be pretty large in this example. And then you have the spatial variance, which is a little smaller. In this case, it's the exact opposite. Again, it's the same thing. You basically imagine a line where the true variogram or the process would hit a ceiling, and you would have a measurement error, a measurement error variance, tau squared. The variance is tau squared in our notation. And then you have sigma squared, which is a spatial variance. Now it's almost 50-50. So that's basically what happens. Okay, so that's stationary, but you do not have to assume isotropic, because you can leave the distance between the sides different. You could even do geometric anisotropy, meaning rotate these using a linear transformation. All sorts of model extensions can be done. But isotropic models are popular because of their simplicity, their interpretability, and because a number of relative simple parametric forms, as I showed you in the previous slide, are all available to us. So let's take a close look at one of them, okay? So here is the covariance function. So if t is equal to 0, it is sigma tau squared plus sigma squared. And if t is bigger than 0, then you have a spatial variance times e to the power minus of phi t. Mind you, this is usually modeling the w. And what is the w? It's here. The tau squared comes from here, and that's always present, all right? Okay, so what we do here is we define the effective range, T naught, as the distance at which this correlation has dropped to only 0.05. Now, why 0.05? Why not 0.01, 0.03? Of course, it could be anything. It's just that 0.05 is somehow treated as a holy value in statistics, and they use that. But as Bayesians, we don't obsess about that at all. You can use a threshold that you like. Setting the exponential function to this value gives us basically this argument, 3 divided by 5, because log of 0.05 is 3, as the effective range parameter. That means beyond this distance, whatever the value of 3 over phi is, the spatial association or the spatial correlation is expected to drop down or dissipate. Then the partial CIL, as I said, is viewed as a spatial effect variance, which is sigma squared. And then sigma squared plus tau squared gives the maximum total variance, which you can call the ceiling or the CIL, or the short form is SILL. The discontinuity at 0, there is a discontinuity here, because if you take the limit of t going to 0, this becomes 1. And so there is sigma squared, whereas the discontinuity here is coming from this tau squared. And that is because of the nugget. And oftentimes it is intentionally put in as a modeler because you want to account for measurement error or microscale variability. And it also lends a certain level of numerical robustness to your matrices. So the empirical semi-variogram, coming back to that, if you recall, is of this form, right? Building a grid of bins or a collection of bins, looking at the squared differences between points within the bin, averaging them. Now, for a stationary Gaussian process, all right, basically this guy, this variogram or semi-variogram, essentially is simply the value of the covariance function at 0 minus the value of the covariance function at a separation of h. And gamma h that you get out of here as a function only of h is the semi-variogram corresponding to the covariance function c of h. So for an exponential GP, if I have given you the covariance function like this, you can also calculate the variogram easily. And so here you have a classic example where a semi-variogram is in fact informing us of spatial structure. Now, even though there is a bit of a cycle here, there are dips here, you notice that the semi-variogram definitely tell us or show a pattern that this is there is spatial structure here, and it's worth investing in spatial modeling and the spatial regression, so spatial effects essentially. So again, here, if you notice for this one, again, this represents this jump here at the tip of the variogram represents the nugget. That's the tau squared estimate, whereas the total variance here is almost about above 1.0, approaching 1.2 perhaps. And that actually is way higher, way larger than the nugget. So the spatial structure dominates. The semi-variogram provides us with a beautiful exploratory data analysis tool to try to get to these estimates. And then here comes the range. If you notice where it is obviously asymptotic. That is fine, but you get an idea about where most of the flatness is, and that can be actually calculated from here. Now, are you really going to be looking at a lot, like days and days of work into trying to explore spatial data? Well, I don't think anybody has that sort of time, because spatial data is way more complicated than time series data, which operates in one dimension. So you might want to actually settle down for a covariance function, because after all, that's a bit of a modeler's choice, and we will do that. The only point I will try to make here is that you don't need to look and explore across a wide array of covariance functions. Just restrict yourself to what is called the Matern family. Now, the Matern family is a complicated family, but it's computable and it's already pre-programmed in R. But let's take a quick look at this. Again, at t equal to 0, it's tau squared plus sigma squared, because that's what the process is, basically the value is. But then here, when you look at it, it's basically where it is in fact going down. So sigma squared divided by 2 to the power nu minus 1 gamma nu. Then here you have 2 square root of nu t times phi raised to the power nu. And then you have a modified Bessel function of order 2 taking on the expansion of, taking on this expansion. It actually can be expanded using a Taylor series. Okay, so what is remarkable, really remarkable about this is this additional parameter. You see, we had only two parameters here. Well, actually 3 parameters, I'm sorry, tau squared, sigma squared, and phi. Here, we will have tau squared, sigma squared, phi, right here, the decay, but also this nu. And nu is in fact a smoothness parameter that controls how smooth the process is. And you could put a little bit of a prior on it and try to learn about it from the data. So nu equal to half, for example, gives you the exponential covariance function. If you equate it to half, you will get a special case. Okay. Now, once you have decided on a spatial covariance function, our next goal is to, in fact, look at the random effects and then try to see how we can use that to do predictions. Okay. So if W of S is modeled as a Gaussian process, just tack on the observation to the observations, the value at location S0. And now your W S0, given the W, it follows a normal distribution with a mean at M of S0 plus C transpose C inverse W minus M, where M is expected W, C is covariance of W, and lowercase c is covariance of W with W S0. The variance comes from what is known as the Schur's complement in statistics. Evaluate the covariance function at the point S0 and S with itself, S0, and then simply take a Schur's complement, C transpose C inverse C, and subtract it from that. So the Gaussian process formulation gives, in fact, the full predictive distribution of WS0 given W. So in other words, the probability of predicting WS0 given a new location or WS0 is actually given by this full predictive distribution. You can compute it using MCMC sampling and so on. I'll mention a little bit about that in the coming segments. So let's summarize this. What have we achieved? I haven't spoken much about Bayesian yet, but we have come a long way. We have, in fact, now a spatial linear model. You have an outcome. You have the linear regression term with regression coefficient beta. You have the spatial process, as we like to call it. And then you have a measurement error or a microscale variability process. So the W of S is modeled as a Gaussian process with 0, C, and theta. The epsilon S is modeled with normal 0 and tau squared contributing to the nugget. The isotropy here is interesting. The W here is a vector of random effects with a multivariate normal distribution. That's what a Gaussian process does. The Gaussian process uses that covariance function here to model the matrix elements as the value of the correlation function between Si and Sj with a given decay parameter and a partial SIL parameter or the spatial variance parameter. So in the end, when you do everything, you get a normal distribution for your data with a regression structure X beta. That's typical, right? And then you have this particular covariance matrix, sigma squared times R of phi plus tau squared of the identity matrix. That's a marginal variance. All right. Now, how do we do parameter estimation? So this is the model, but we don't know the beta. We don't know the values of WS at different locations, right? And we don't know the variance of this epsilon S, the variance of the W, and everything else that has gone into the specification of W. So that means sigma squared and phi. So we have to estimate that. That's what a model does, right? Whether you're in machine learning or in statistics, you're going to try to use the model to try to get an estimate of the parameters, because those parameters can then be plugged into the model, and then you can probably do some inference, do predictions with the fitted model, and so on. So we can obtain the maximum likelihood estimates of these parameters based on the above model, and then we can use these estimates to Krieg, meaning at S0, we can easily find Y of S0. That is entirely possible. There are the Krieging equations, which basically look at the conditional expectation of Y of S0 given Y. Nowadays, there's so much software, you can easily do that on your own. This is almost exploratory data analysis, unless you are looking at the variability in the MLEs and the variance structure and so on. In practice, the likelihood is often very flat with respect to the spatial covariance parameters. So if you fix everything else, but either look at phi or if you look at sigma squared, this likelihood is usually quite flat. And that actually does no help to the information section here. So in terms of inference, in terms of the finding, it is of no help in trying to get to the parameters. Okay, so that's why these parameters, sigma squared, phi, and tau squared, we say are weakly identified by the data. So they're not very well identified by the data because the likelihood will struggle. So what we usually do in practice is run an empirical semi-variogram of the residuals using an ordinary linear regression, and then try to fix some of these parameters, focus on estimating the beta, maybe the nugget perhaps, and then use those values to in fact do predictions or spatial interpolation, or what is commonly known as Krieging. For spatial data, there are metrics that you can use to actually assess the goodness of fit of your model, or even look at exploratory metrics that will tell you whether the model is performing as well as you want it to. So in terms of model comparisons, you have two very interesting information criteria, AIC and BIC, the Akaike and the Bayesian. There's really not much Bayesian about it. In fact, Bayesians use the WAIC, which is the widely applicable information criteria more often. They're all coded up in R, so you can actually calculate them quite easily using packages. But then, you have the prediction-based performances where you can look at holdout data, which is simply like a variance of the holdout data. So here is your holdout data yi. This is how your trained model will predict the holdout data. And then you square the distance and then simply take the average and take the square root finally. Then you can look at coverage probability. So you would have the prediction intervals that you will compute. And then basically you see how many random variables essentially are in this, and you can report that. Or you can look at the width of the 95% confidence interval. And that basically is simply the value here. You might want to take the absolute value because if it's negative, it won't make sense. So absolute value is fine. The last two approaches, that is the coverage probability and this guy, compares the distribution of y instead of comparing just their point prediction. So you see here, it's just a point prediction. Here, you see it's just the estimated predictions. But the current approach here, where this and this, the coverage as well as the width of the 95% confidence interval, are two metrics which tend to use the quantiles of the data, as in this case, or in this case, or more generally, it can compare the distributions in terms of the joint predictive ability. And so not everything is bad about this, and they work quite well in practice. So here's an example. If you look at the spatial data that Andrew had, you can look at the AIC and the BIC. They still do a little better than the non-spatial model, but where they, yeah, than the non-spatial model, but the root mean square prediction error is not a whole lot lower. The coverage probabilities are not a whole lot different. In fact, to a certain decimal point, they're probably both 93 here. And then you look at the confidence interval width, and the spatial definitely shrinks the length of the confidence interval a little bit. Now, we didn't do the WIC here, but in most of our papers, we actually compute the WIC. So here you have the DBH estimates, and then here you have the standard errors. And so as you can see, the standard errors are higher, tend to be higher where there are there's more data available, and that can be helped. So in summary, before we take our break in a minute or so, we have geostatistics as the analysis of point reference spatial data. We look at surface plots of data and residuals. We look at the EDA, exploratory data analysis with empirical semi-variograms. We look at modeling the unknown surfaces with Gaussian processes. We look at kriging, which is predictions at new locations. And we have looked at spatial linear regression using Gaussian processes. So we've covered a fair bit of an overview, but even if you don't want to get into the details of this, the one important message in terms of spatial statistics here from the first hour of this short course that you should take with you is that not all data really needs spatial random effects. And so we need to look at empirical data science or empirical exploratory data analysis, EDA, to identify certain parameters, see the ease of estimation by fixing some parameters, and then moving on with prediction and so on. So there's a graduation. You always start with EDA, construct the variogram, compute the variogram, read off through eyeballs, or otherwise optimize the variogram. Those can be done in our packages, and get the values of the hyperparameters that reside in the spatial covariance function. And then fix those values, and then suddenly you are talking business again. All right. I think that ends the first segment. So let's take a quick break for 10 minutes, and then maybe I will come back and give a little computer demo. Are there any questions? Feel free to interrupt me also if you have questions. Okay, well, let's take the break.
Yes.
And then we will talk about that.
So Dipto, I had a quick question for you.
Yeah. Go ahead.
Why is the nugget referred to as the measurement error when it seems like it's kind of the non-spatial residual error? Why isn't it just called a residual error?
Yeah, you can absolutely call it, what's in a name.
Yeah, sure.
But I will tell you, it's most of these this nomenclature has come from historical perspectives, and when this is in the 1950s and 60s, even before statisticians, geologists, and those in the mining community were using, like the variograms and so on. And when they saw that phenomena of this blip, they basically felt that there was a process. They also understood processes, not as well or not as formally as we do today, but they understood the process there. And the variogram was supposed to estimate a process. And they felt that that part would be completely explained by measurement error variance or measurement error. That's how that came by. But you're right, it is in fact a residual variance. You could even call it a non-spatial aspect of the variance. And then some people like to look at the ratio of the sigma squared and the tau squared and see which one dominates. And typically in a variogram, because the variogram flattens out, you will notice that if there is indeed spatial, it will take some time to flatten out, and that makes the sigma squared bigger. So there is that analogy as well.
Well, thanks. That's helpful.
Thank you. So we will come back at around 2:18, is that right?
Okay.
Okay.
Yes, I was saying like 2:15, but
2:15 is fine. Yeah, whatever you want. Yeah. Okay, let me grab a quick glass of water and I'll be back.
Thank you.
Okay, let me try to share my screen. Are most of the participants from UCLA?
Most of them are from UCLA.
Okay.
Yeah.
Give me a second.
In fact, our stat group is also attending.
I see. So I believe I can just share the desktop, and that way I believe they will all be able to see everything. Because I need I'll just do a little demo of an R function. So let me. Okay, so this is the part where I think you guys will really be missing Casey, because I am very, very, I would say, not only old-fashioned, but also idiosyncratic in my computing habits. So I was raised in an era before R and so on. So everything to me is the command line. And I still am very comfortable when I do R programs to do everything from the command line. So I don't use like R Studio or anything else. Everything is coded up in scripts. Now, of course, you guys can probably do much better than me. And I'm sure Casey gave you a lovely tour of our studio in the morning and all the beautiful things that can happen. But what I'll do is I'm just going to tell you exactly how you can code up using the bare minimum in R, a few things to map data. And in the folder, so I can do a few things. I can do a little bit of intro to Geostat, but before that, let's take the first most basic aspect of spatial mapping, as we call it. And let us suppose you have data from different counties in California. All right. And that is exactly what you have in the data set called age-adjusted CSV. and that's also in the folder. And basically it gives you age-adjusted cancer rates, lung cancer rates in the different counties in California. And let's suppose you want to actually use R to just plot that data onto a nice looking map to try to see how it looks like, okay? So before we begin, and by the way, I use a very, I would say, personally tuned Debian Linux operating system that actually is I need for my other research computing needs, high performance computing needs, including connecting to the Hoffman and so on. And I have my C programs and stuff here. So my operating system is also not as pleasant to look at as perhaps your Macs or maybe the modern Windows machines are. So I beg your forgiveness for that. But let's try to get to the crux of the issue, which is basically you need a few packages and then you need a few commands. That's what you need, right? And whatever you use environment, this you cannot escape if you want to do it with R. So the packages that you need are maps, spdep, map tools, class interval, and R color brewer. Maps is a maps database. By the way, I don't want to repeat too much that Casey did or said. Casey, did you do any mapping of geographic data at all in your morning session?
Nope, no mapping at all.
Okay, good. So that's good. I didn't want to necessarily do that. So the maps database is a very rich database inbuilt in R through the maps library. That all of this, by the way, you can download from CRAN. Essentially what you can do is you can look at the maps database and you can extract a county map for California. So here, take a look at this first segment right here. All right. What I'm doing, I won't go line by line. That is something I do in great detail when I teach the whole quarter spatial course. I try to go under the hood and try to explain the polygonal data structures and rasters and so on. I really cannot do that here because I would like to give you more of an overview of this. But what I'm doing here essentially is this, that I'm looking at the map function inside the maps library, looking at the region of California and saying that I'm basically extracting from its database, from the maps is nothing but a big database, a county map of California. So if I just take this, copy and paste this into the R terminal, all right, let's do that. Can you guys see both my my program and also this terminal? Because I will have to, as I said, I don't use the fancy R stuff, so I just run R from the terminal. But you can see this, right?
Yes, we can see that.
Okay, good. Oh, I'm sorry. Oh, I'm sorry. I forgot to load the libraries. There you go. All right, here you go. I will first load the libraries. And let me give a little space here so that we can see this. And then what I'm going to do is I'm going to simply take the county map for California and see how that looks. Now see, right now there are no maps, because what I've done is I have actually just extracted the coordinates and the polygons for the counties. The polygons basically means the line segments. Every map database is nothing but comprises line segments, very small perhaps, that essentially approximate boundaries like coastlines or polygonal state boundaries, county boundaries, something like that. All I'm doing here is simply taking out California and the county polygons. And now, what I do is the important data object here, the map object, is the polygon data frame, which is basically created from the map to spatial polygons. Spatial polygons are a very important part of the R spatial packages. And it's something that you can Google and you can find out much more about, or you can take my class when I offer it. But basically, once I have the California.poly, which I created, this polygonal structure, then I can plot it and I can provide, maybe the red boundaries and color them red, or do fancy stuff with it. So right here, here you go. You have the R graphics. That's the plot of California. Now, if you want to color the boundaries, or rather the labels, and if you want to color the labels red, right here, you have the names and they all appear in red. Okay. So this is GIS for you as an R. R is offering you GIS. This is exactly what a ArcView or one of these paid products do. They extract a map for you from some part of the world, and R can do that for you completely free. Okay, so that's beautiful. All right, now this is good. This is already good because we don't need a separate separate software to do our mapping or our GIS, but it's not that useful yet because it's just extracting data from what is already inbuilt in R, the maps library. Obviously, as researchers and data analysts, you want to actually plot data that will be provided to you, or that you yourself will collect or maybe extract from somewhere else, right? So in order to do that, you take a look at the for example, this segment here, the second segment. What we'll do is we'll now plot data on the map. Now this takes a little bit of getting used to. Why? Because usually in the ArcGIS or those types of software, it's just everything is point and click. Whereas here, even I believe with RStudio, you still need to write some scripts. And so here's what I do. I look at I read, first of all, the data, the 5-year lung cancer rates from each of the counties of California. Then basically what I do is I essentially, sorry, the first line here, let me go line by line. I don't want to skip too much, but I don't want to go into too much detail. Right here, I'm reading the data from my CSV file, which is basically converted from an Excel spreadsheet. And it gives you the 5-year rates, all right, from all the states across the United States. Then what I do is I am going to look at using this. Now, if you go into this particular database, this particular Excel spreadsheet, you will see why I'm doing this. But I'm basically looking at this variable, the state, county, and then I'm looking at the column numbers and extracting simply the part from California. So this is called a substrata function that I can use here, and I can look at that. And then, I further basically look at the California rates, and then I extract lung and bronchus, which is basically lung cancer. And then I look at the rate of lung cancer, where I actually combine essentially what I order it in a way so that I can look at the county and this variable. By this variable, I'm going to prepare to merge these rates with what I have from the maps database, because I don't want my counties to be mismatched. The counties from this data set that I read, I need that to be aligned with the counties here. Otherwise, it's going to do it in a different order. So you see this order function, and with a little bit of a parsing, can help us basically get the data in the right order in terms of the counties in the map database, so that I will be able to plot the right rates corresponding to the right counties. So there's that little step that is very important. Otherwise, you'll get a completely incorrect map. And then finally, you can import the lung cancer data in the map data simply using this. And then the last part is about doing some nice plots. Okay, that's the idea of this. So let's take a look at the second segment. Let's run that. Okay, so the data processing is done. Now I'm going to get to the plots, but in order to prepare for the plots, I want to basically take a look at actually, I did a little bit of EDA here in terms of trying to see where to cut off the what is called the choropleth map, essentially the bar that distinguishes between the different colors. So I am looking at 5 different segments, which I'm going to use in 4 different colors, shade from red to blue. So let's see how that looks like. Okay, that's where the plots come in. And I'm going to create this, and then I'm going to plot, add some legends, and I'll be done. So bingo, here you go. And finally, when I can add a legend to make it look nice right here. So the color coding corresponds to the different values. So anything between 22 and 41 is going to be shaded here. 41 to 45 will be shaded here. And you see how there are the 4 segments. Now, where did I specify that? If you're wondering where does this 22 41 come in, right here. You see, I create this breaks sort of at 22, starting at 22, 41, 45, 51, 80, right? That's how it actually goes. So here are your 4 categories, and I'm using these 4 color shades to do that. Okay, so this is the very, very basic code that you can use. For example, if you have county level data in the United States, or you could change it, and then there's a lot more that can be done in R. If somebody gives you their own shapefile, then you can actually convert that into a map, get data, plot it. These are things that we teach in our spatial class in biostatistics, and there's a lot that can be done only with the plotting and the mapping features available to us through the different libraries in R. All right. Now, once that is done, I want to move to a different program. So let's see, which is the intro geostat EDA.R. Now, this is actually a little bit, hang on, let me close this window. This is a little bit more aligned to exactly what I was talking about, variograms and stuff, and how we can do that. So this is a very long program because this has a lot of examples. So let me see how much I can do here. But let us suppose, actually, I can't, maybe what I will do is, let me strategize here a little. Okay, fine. Let's first of all start with the packages. By the way, a very good habit, if you're writing an R program that you want to do from source, meaning that it will be a standalone program, is to actually start with this this little snippet of code. What it does is it removes pretty much everything from the R base. Otherwise, if you have an error in your coding where you have a variable that is already in the workspace, you could get confused and you could run into errors that may be difficult to debug. So that's something I've learned from experience, and I like to do that. Okay, now here is your the different libraries, the MBA, the fields, the class interval, GOR. So, oh yeah, TV, go ahead.
So I was wondering, what's the max resolution on these maps? Could it be like, for example, UCLA buildings could be mapped on it? No?
Yeah, for that, not from the maps database, but there are other R. I think it's called R Earth, and there used to be an R Google Maps package, but I think they ran into a little bit of copyright issue with which you could have done that. I don't know if our Google Maps is still available from CRAN, but you can surely download it as a separate entity, and you can look at that. And then you basically have all the features that Google Maps gives you. So you can go down to your office
Oh, okay. Thank you.
Yeah, you're welcome. Okay, so let's try this. I'll download some of the packages that I need. So these are the very basic packages that you need: MBA fields, class interval. We already saw that before. This one is used to create those breaks in your color codes and stuff. So let's try to take a look at what we have as data set number 1 here. And I'm going to go a little faster and do blocks here. Otherwise, it'll take me a very long time. So what I'm going to do here is first create a little myplot function that actually does nothing but streamlines my plotting. That's pretty much what I will do. All right. So let us take a look at until this point and see what we have. Okay, here you go. So this is your Y. Remember the slides I showed you? This is basically that program that generates that. You have a data generated. It's called dataset 1. It's in the data folder, which is also there in that zip folder that Casey has generously linked in his GitHub. And you could easily identify this. This is what I was talking about, right? Here's a covariate. The covariate has a pattern. The Y has a pattern. Clearly, they go the other way. So there's a negative association here. So that's what it is. And then, if I look at this guy just fits a linear regression, ordinary linear regression, and collects the residuals. So what I'm going to do is I'm going to fit a linear regression. I'm going to collect my residuals, and then I'm going to plot. Do not try to do this with me, because if you fall behind, I won't have time to really go back and individually cater to it. You can always email me later on. The code is there. The only thing I guess you might need is that this code is adapted to my machine. You may want to actually see if it needs any adapting or how portable it is. So that's the only thing that perhaps is of value or worth taking a note of. So let's take a look at the next segment. And here, if you recall, we saw that this is the random residuals, not really a lot of spatial data. So therefore, if there's not a lot of spatial data, what we do is, I'm going to basically skip the variogram part for that, okay? Because it won't look good. It's the same one in the thing. Let's come to the Oregon data, the Western Experimental Forest data. That's the Oregon forest, and that actually has the species and stuff. So how did I create some of those plots for species and so on? So let me close some of these windows, because otherwise I'll get cluttered and I won't be able to manage all of them. All right. So if you look here, I'm going to read the Western experimental data from the CSV file that's also in the data folder. And I'm also going to hold out some data for predictions. And I'm going to free up some space because I'm going to use a smaller data set. As you can see, I'm using a smaller data set, and I'm only extracting 2 different pieces, one for training and one for holdout. So I'll do that little piece here. And then I'm also going to get to the variables, extract the variables, and then I'm also going to create a color ramp palette. Okay, I'm doing a lot of things. Sorry, I can't go line by line. It'll take too long. But you basically, the gist is processing the data here. All right, no statistics going on at this point, but extracting data and using R to try to provide some exploratory data analysis for your spatial data set. All right, so let's take a quick look here. So we have extracted pretty much everything, no errors. Okay, good. So that is done. Now we come to the quantiles-based color coding of the location. So we'll see, this is where, these, the values can in fact be looked at by quantiles, and we will color code them. So let's take a quick look at this part because you will see an interesting plot here. I can't remember if I showed this to you in the in my slide, but let's take a quick look. Right here, there you go. So this one basically shows you the locations, and it basically gives you the values in terms of the quantiles, which is color coded like this, as to where the values are. So as you can see, deep blue is the smallest dBH, that means thin trunks, slim trunks, and red are the the trunks that are pretty thick and fat. So that's the way. I couldn't use the word thick because thickness is something quite different, but it's kind of like how much the circumference of the trunk is, about 4 meters from the ground. That's what basically it does. And it's okay to look at these plots. They do tell you a little bit about where the trees with low dBH are, where the trees with high dBH are, but you can see they're pretty much all scattered. So we'll try to see a few other alternative plots here. One that I really like are these surface plots, which essentially does a surface interpolation, fully deterministic, no inference, so to speak, but good exploratory plots. So let's try that with the MBA package and see if that works. There you go. This is, I think I showed this to you there. You create a surface basically using this line here, this function mba.surf, that actually creates the surface with the coordinates and the value of the dBH. It's pretty easy. You don't have to worry too much about this h equal to 5, m equal to 2. That's part of the algorithm that determines it. It basically uses a hierarchical B-spline to interpolate with no uncertainty quantification at all. So purely deterministic, but it does a little bit of smoothing depending upon how many hierarchies you have. If you reduce the number of hierarchies, then you will basically reduce the smoothing and so on. All right. Then let's take a look at the plot of the species. Let me again clear this out for a little bit. And let's take a look at how we plot the species. So here again, after you have created, so here was a quantile plot, right? This was a quantile plot. We just created the class intervals. This is where the class int package comes in. Look at dBH, look at the number of classifications you want, and what do you want it by? How do you want it classified? By quantile. Fairly intuitive. And then you use the find colors. This comes from the R color brewer package. You have the color palette has already been created here. Use the same color palette, and then simply plot the quantiles. And this PCH, CX, these control the size of the circles there. You can make it bigger or smaller. You can choose. You have a lot of control. If you like coding, you will like this. You can control your plots. So R is a language based on the S language, which is essentially designed to take data as objects, right? And so it's a language for data. That's why it has made such a massive comeback in data science. It was literally used only by statisticians for many years, but then folks who are now doing data science realize its advantages, and together with Python, I believe it really occupies a fair bit of the space in data science. Okay. So here, let's look at the legends on terms of the species type. So these are the 4 species that we're looking at, and we try to see where the species are. So you can see a little bit of the clustering of DF. I forget what that one is. I think the data file will tell you what it is. But the different color codes, in fact, you can see some clustering of by species. So this is useful. You basically try to see how the species are scattered. All right, that gives you that information. And then you come to a little bit of the statistics here. You look at the residuals, you fit a linear regression model, look at the residuals, and let's plot the residuals. All right, so we will do that. That's fairly straightforward. Just get the residuals from your LM and then simply plot it by the surface interpolator using the mba.surf, and that'll just work fine. There you go. Remember this picture? This is so much of spatial clustering. Okay, you see, it tends to basically say that there's a lot of residual structure in the space. Now, if you're an epidemiologist or someone, and this was a disease map, you would start hypothesizing, wait a minute, why is it like this? Why is this blue here? Why is this red here? Can we look at lurking covariates? And sometimes in forestry, we do that as well. But in forestry, the more important thing is to account for this variability and develop a better predictive framework, because prediction is of greater importance. So it's a lot of predictive analytics that goes on. Okay, so we can look at the variograms for the raw data. I suppose I'm going to close a couple of these windows, otherwise there'll be so many windows I won't know which one is which. All right, here you go. You have the variance. Hang on for a second. Okay, let me get rid of this one as well. This one is the distance, the semivariance of the raw, the residuals. No, sorry, the which one was this? This was the, let's see what we did here. Oh yeah, this is for the DBH, the first one. And then the second one here is for the residuals. So we can do that in R fairly straightforward using the GOR package. The variog function does the variogram. If you're doing spatial point reference data analysis, the variogram function is a very critical function. So if you're interested in this, spend some time in exploring this function. In the GOR package, there's another package called gstat, which also offers pretty much the same functionality. All right, then you can come to the spatial maximum likelihood estimates, and you can actually use the likelihood fitting function in GOR, and you can also calculate AIC and BIC to see how things are working out as comparing the AIC and BIC between the spatial model and the non-spatial model. So give it a few seconds because it will need to converge, but here you go. So as you can see, with the spatial model, the AIC scores are quite a bit lower than their corresponding BIC, the corresponding non-spatial scores. So you compare 4466 with 4508. There's not much in comparing this guy with this guy because they're basically calculating two different quantities. But look at the AIC for the spatial model, which is here, as opposed to the non-spatial model, and you see you get about what about 34, 42 points lower, right? And then similarly, the BIC also is a fairly lower than the non-spatial BIC. So the lower these information criteria, the better the fit. So you can clearly see that what we were intuiting from those graphs about there being spatial structure is now being formally established using some numbers and some statistical theory about information criteria and goodness of model fit. Now you could look at other nice things like root mean square prediction error. Let's see, what's the time? Yeah, I'll probably finish this segment and then go for another little break. Just give me a second here. All right, okay, so I've closed everything. So let's take a look at the root mean square prediction error. So how do we do this? Again, very quickly, you actually use the Krigg. So that means you're doing the spatial modeling and you're Krigging, which means you're doing predictions, okay? So this is model-based spatial prediction here. This conv doesn't stand for convergence, it stands for conventional, meaning as opposed to Bayesian. I'll talk about Bayesian in the last segment. So you basically look at this guy, then you can do the prediction using the maximum likelihood estimate fit at the predicted locations. Then you basically look at the fit the predictive values and look at the root mean square error. That's basically the sequence. You can probably understand this sequence fairly clearly once you go through the details and compare with my slide here. So let me just go for a second backward right here. This is the second segment that I'm now going to compute. This was done previously. There may be a little bit of difference between what I'm doing here and here because the data set that I might have used for this is a little different, but the story pretty much remains the same. So let's just do that. So here you go for the spatial, you see it's about 18.9 or 19. For the maximum likelihood estimates, it's about 21. Now, this is based only, I think, in about 50 holdout locations, if I'm not mistaken, or maybe I am mistaken. But approximately, as you can see, it's about the same as for this particular data set, whatever this was. And the question that you might want to ask is, well, how do I know that whether this is bigger or not? I mean, this is big enough to make a difference. And you're right, you know, for the RMSP, it's a little difficult to actually calibrate this thing. But for AIC and BIC, typically, you know, if you're getting a 10-15% reduction, it basically means that you're definitely doing better in terms of goodness of fit. So if you have the resources, go spatial over the non-spatial, at least for this example. And then finally, you know, there are a few other metrics that we can look at, like the CP, which is the coverage probability. So that means how much are we actually coverage? What is the coverage of including the true values in the prediction intervals that we have, and then the confidence interval width. So those can also be calculated right here. So here is the confidence intervals that you calculate, and you can actually easily calculate the confidence interval width using the mean confidence interval. Because remember, the confidence interval width is actually calculated for each of the random effects for each of the locations, right? So one of the things that you can do is you can calculate the width for each one at each location and then simply take the average. So that's the spatial and the non-spatial aspects. And if you just, again, simply run this segment, you get about the credible intervals. That's that part. Here's the spatial part, 0.97, slightly wider than the linear model. That's okay, because you know it doesn't really make much of a difference here. Including the spatial random effects can in fact increase the width a little bit. And then CIW is about 80 for the spatial, that's the confidence interval width, whereas the confidence interval width from the linear regression is about 85. But the coverage is about 97 and 95, which is pretty much similar. And then finally, you know, here's how you can do a predictive surface. This is important because what we do is we interpolate over the entire domain. This is statistical interpolation as opposed to deterministic interpolation. And so we are using the estimates from our statistical model now to actually create the maps. And so that actually is, let's take a quick look here. And I guess in RStudio, you can just source this in the same environment, which is advantageous. I haven't bothered to install RStudio. Oops, something happened here. Give me a second. So here is the predictive map that you see, which in fact looks at the prediction values or the spatial interpolation using the model. And there are two different plots that we look at. One is the predicted value of the residual plots. So spatial residuals right here, whereas the previous plot was that for the interpolated values. So this is for the W in my notation. So if you go back here, there are the two plots. Oh, sorry, I'm sorry, this is a standard errors. I made a mistake. Let me double check. Yeah, it is a standard error because I'm taking the square root of the predicted variance. My mistake, not the predicted value, but the standard errors. So here is the standard errors of the predictions. The previous plot was the value of the predicted of the DBH estimates from the model fitted estimates and standard errors. All right, so that actually concludes this segment of Introduction to Geostatistics. And you know what I'm thinking, TV, is that let's take the break now, because that'll be a good time to just come back and get into the Bayesian part. That will be the last segment. Is that okay, TV? Oh, you're muted. I'm sorry.
Yeah, 3:05 then?
Yeah, that'll be good.
Okay.
All right. So let me get into the last segment where, so until now, you know, we have actually done exploratory data analysis, basically giving you the overview of the sort of spatial data science, as I like to call it. And then a little bit of statistical modeling development in terms of trying to smooth spatial effects, trying to understand you know, how model-based prediction works and how that can be implemented in R. So this last segment I'm going to devote to the Bayesian modeling and Bayesian geostatistics. And depending upon how we do, we have a stop at 4 p.m. I'll see to what extent I can do a simulation example or something like that. All right. So this is the slide called Bayesian_geostatistics.pdf, and that's in your packet. So let me see, I will do this as a. So spring 2021 was the last time I taught that spatial course, by the way. So these are all snippets of slides from that course, so that I'm using. I'm not offering that course this quarter in spring 22, but it will come back in spring 2023. So let's take a quick look. This may look a little, you know, daunting at first with all these notations, but it's really not very difficult if you go line by line. So first of all, we need to basically look at what we call the Bayesian spatial random effect models. Okay, this is very similar to what essentially random effect models are. It's the thinking that is a little different in terms of how you want to construct the model. And in Bayes, what we like to do is we like to think in terms of hierarchies or in terms of steps. So in the first step, we look at the data and we say that conditional on some mean function and some measurement error variance, or as Casey says, residual variance, let's assume that it follows a normal distribution with that mean function mu of SI and the residual spatial variance tau squared. Okay, each data point, that corresponds to each spatial location follows this. Then the second stage, we deterministically say what this mean function is. Deterministic, in general, in a linear regression, up to parameters, right, like betas and the x's, but then in random effect models, up to not only the regression coefficient parameters, but also the spatial random effect values. So in other words, you see, what I'm trying to do is I'm saying that, look, if you give me a couple of parameters here, like the mean function and the tau squared, I specify a distribution. Then if you give me a value of a random effect, and if you give me a value of the betas, then, or the slopes, regression slopes, with values of prediction, with predictors or covariates, then I know exactly what this mean function is going to be. And then comes the priors. So basically, who gets the priors? Essentially everything that is unknown. So what are the unknowns in the first two stages? Under this parameterization, the unknowns are the betas and the W's. So the betas, you give a prior. You could give them independent. Each beta, you can give an independent normal 0 sigma squared beta prior. And by the way, this sigma squared beta could even be infinity, literally in the calculus sense, so that this becomes what is called an improper prior. That's also fine in these sorts of models. That would reflect no prior information at all on the regression slopes, which is what classical statistics in any case assumes. But here we need some help with regard to the process, because the process, if you remember, is a Gaussian process. So how does that translate to this hierarchical model? Well, collect the process realization is what we call it. So that is the value of the random effects from S1 through Sn. Collect them into a W, a random vector, and simply, because it's a Gaussian process, you put in a spatial correlation matrix. I'm going to come to that in a second, but that's what it does. And then finally, we put to the any variance components usually get an inverse gamma distribution, which basically means 1 over the variance components get gamma distributions. And you can put in some priors here, like values like 0.1, 0.1, 0.01, 0.01. Usually the data will overwhelm the priors and you won't really get a lot of sensitivity to these values, trust me. And then finally, anything that is inside this correlation matrix will get something like a uniform prior. So this actually is a first example of a spatial random effects hierarchical model. As you can see, there are only two important things here. One is inserting the spatial random effect into the regression component, and then secondly, putting the prior on the spatial random effect. And I believe Casey did his STAN examples in the morning. You basically take the model, think exactly like this. It'll make life much easier if you just translate it into the language of STAN. Okay, now here comes an important part. This correlation matrix, how is this formed? We saw this in the previous segment, but you may or may not have realized its importance, which is, remember that table for covariance functions? Take any such function and evaluate it between the pairs of locations here. So this gives you, you see there are n locations. So each element is evaluating a correlation matrix. Remember there was this parameter phi in the exponential, for example. There's the range parameter, or the inverse of the range parameter, or the decay parameter, you would call it. And you evaluate that at the pairs of locations Si, Sj, and that gives you the ijth element. And then this is the n by n spatial correlation matrix. For count data, you have, this is a Poisson with an intensity function lambda. The log of lambda gets this specification. And then here you have the betas, and they get their priors. Same priors for the spatial process, no difference. because we are using a GLM or a generalized linear model, this is the link function, right? It's Poisson regression. So this can still be scaled to a normal, and therefore a GP will still work, and you put that in. The only difference here is that this tau squared, which is an additional variance measurement or variance parameter in the Poisson, doesn't come in because the Poisson's intensity function itself takes care of the first stage distribution. Now, some papers and some researchers like to tag in a noise here, an independent random effect here after this, and bring in the tau square here into here. I personally have never been a big fan of doing that. There are some technical reasons for that, but there are some practical reasons for that as well. The technical reason is that it introduces a discontinuity in the surface, which essentially could lead to some MCMC issues. But the practical reason is, you see, this is actually what is, when you look at a Poisson variable, like a count variable, okay, let's say you're looking at the number of trees in a particular pixel, okay, so SI is a little square area, small area, and you look at the number of trees there, okay. But what you're looking at the intensity is like a Poisson process. And so therefore the intensity basically tells you the average number of trees there would be this. And what the space does, the spatial effect does, is says that if you are near me, for a pixel that is near another pixel, the expected values of the number of trees, or the intensity that we are observing in the language of Poisson regression, is going to be of similar value. It's not necessary that the count itself is of similar value, but it's the expected intensity that is of similar value. And in that case, why should the intensity or the average value, which is a completely latent thing, in fact get a noise, right? Noise usually comes either from measurement error or some microscale variation. Now, if you have microscale variation, perhaps there's some point in putting that in, but that would mean that you would need some information about locations at very close points, right? Pairs of locations that you have not observed. So it's a bit tricky whether you want to put it or not. It's not wrong necessarily, but whether it is useful or not is questionable. The rest of the specification remains the same. So when we look at the W's in both these cases, binary data, sorry, I missed one slide. This is very useful, logistic spatial regression, right? Useful for classification as well. So you look at the Y's, they're 0 or 1, let's say rainfall at a location or no rainfall, right? And you look at a Bernoulli distribution, and what you're modeling is the probability. So in other words, the spatial binary model will say that if 2 pixels or 2 locations are close by, then the probability of them seeing rain are similar, and the probability of them seeing no rain are similar. So in other words, if you have a 1, a neighbor is more likely to have a 1. That does not mean that neighbor will have a 1. You could have 1 in a location and you could have a 0 very much in the data set in the next location. But the borrowing of strength comes from this Tobler's law, as we call it, which is modeled using a Gaussian process. The rest of this is pretty similar. So now here, again, just to remind you, the WS gets a Gaussian process. So the multivariate normal comes in here. This becomes a sigma squared times rho. That was what I was calling C in my previous lecture, in that thing. So now I separate the variance with the correlation, and I use the correlation function to model the elements. So here's a very practical choice. The first law of differential equations, for if you have taken calculus, you will remember this. Basically, the exponential decay, right? So in other words, the decay is proportional to the distance between those guys. The rate of decay, in fact, is basically a constant times the current value of the correlation, and that's a valid correlation function. There is a bridge theory here. This, you cannot dream about a function and bring this in here, because this matrix may no longer be positive definite, which will translate into your R code with an error saying that the Cholesky decomposition has failed. Because only positive definite matrices allow Cholesky decompositions, and you will not be able to run your model unless it's positive definite. So there's a theory, very rich theory in mathematics, on positive definite functions. What are those such functions? Such functions are those functions that when you plug them in a matrix and compute the different values of that function for pairs of locations, the resulting matrix will be positive definite, which basically means the resulting matrix will have eigenvalues that are positive or will admit a Cholesky decomposition. There are lots of different characterizations of that sort of thing. Okay. So how do we actually estimate now, right? That's a big question. So this is the model construction, and how are we going to estimate? So here you need some form of MCMC. Now there's a lot of variability here. You could use Gibbs samplers, you could use Metropolis algorithms, you could use Hamiltonian Monte Carlo, which is what Stan uses. And you guys have all seen Stan in your previous, in the morning from Casey. So here, the idea is that when you think about Bayes, you think like this, that I will be essentially looking at the posterior, which is necessarily the probability of the unknowns given the knowns. And in each of these models, depending upon the specific parameterization, we're looking at the normal model here. You have the regression coefficients, you have the spatial random effects, you have the two variance components, tau squared for the measurement, or the residual, I should say, after Casey asked me that question, and the spatial variance component and the range. These are all unknown. So it's the probability of the unknown given what is known. What is the data? It's the y, which you have observed as the dependent variable, and the x, which is the matrix of predictors. So you estimate the latent process at a new location, and you sample from WS0 given W and sigma squared and phi. And then you obtain the posterior samples of mu of S0 through this. And you predict y of S0 by drawing its value from here. So each sampled mu of S0 coming from step 3, and then tau squared from step 1, prediction is simply going to be drawing a y from a normal. Now this part, if you're predicting within Stan, it will do exactly this. This is the part that is different between Stan and OpenBugs. Bugs uses a Gibbs sampler with possibly Metropolis updates for these guys. Stan actually uses Hamiltonian Monte Carlo. So it does not use a Gibbs sampler. It's a different variation of MCMC, and uses Hamiltonian equations to try to work that out. But the more efficient, if you want to use Stan, it may be more efficient to actually do a little bit of mathematics, integrate out this W from this posterior, get the posterior in terms of beta, tau squared, sigma squared, and phi, and then essentially come back and recover the W from its full conditional after convergence of beta, tau squared, sigma squared, and phi, because the W is what is high dimensional. Remember, if you have 1 million locations, you will have 1 million random effects. That is where Stan will struggle. OpenBugs will also struggle unless you use your hard-code, hand-coded algorithm to try to somehow obviate that obstacle, or do some mathematics, integrate that out. It's not hard. You get the marginalized, or what in machine learning is called the collapsed model, and then recover estimate the other parameters, beta, tau squared, sigma squared, and phi given y and x, and then simply draw W from the posterior samples of beta, tau squared, sigma squared, and phi. And that's possible from its full conditional. And that's exact after convergence of these parameters. So it's a very efficient way to do that. But there are other obstacles you do get from big spatial data, which I won't have much time talking about. What happens in the Poisson? Pretty much the same. except the tau squared isn't there. So again, you were going to estimate using MCMC, get the post samples from the posterior, then estimate the latent process by drawing from the full conditional here, and then a latent process at S0, a new location, then simply plug in the values of the betas and the W's here. So you'll get a whole posterior for the lambda, because you'll be plugging in each sample of beta naught and each sample of WS naught. So you'll get a sample of lambda S naught. And then simply for each sample lambda S naught, you draw a YS naught, and that gives you the predictive distribution. Exactly the same here. It's just that the distribution has changed and one additional parameter, but the overall step is the same. And then in the binary, it's also pretty much the same. Again, get your MCMC to get the posterior estimates at a new location, estimate the latent random effects. This one has the W's at any of the observed locations, so you don't have to worry about that. It's a new location that is needed for prediction. So we will get that value here. Then we will use the posterior sample of the P of S naught from here just by plugging in the samples. And then finally drawing YS naught for each sample P of S naught to get the predictive distribution. Pretty much the same steps, just adapted with some minor modifications depending upon the continuous model or the discrete count model or the discrete binary model. So the important question is about setting priors. So for the regression slopes, we customarily assign informative priors. And for the variance component sigma squared, we actually choose an inverse gamma, or equivalently a gamma prior for 1 over sigma squared. The shape parameter is taken to be 2, and the scale parameter is chosen so that the prior mean is equal to the scale parameter. So this value can be set from an exploratory variogram analysis, and the strategy for tau squared is kind of similar. The range parameter needs, which remember gets a uniform prior in the hierarchical model, usually works well. Is a little bit more tricky. You could put a gamma prior on that, but sometimes it doesn't work that well. And so what we do is we usually set it so that the effective range, so that's the distance where the spatial correlation drops to 0.05, is between some small number and does not exceed to about 50% of the maximum intercide distance. So for example, with the exponential correlation function, we solve for the distance where the correlation drops to 0.05 or below. And then we see that phi is approximately, from this equation, e to the power minus phi d in particular will give you approximately 3 over d. So d is the effective range. So if we bound d between effective range, let's say 35 to 40% or 40 to 50% of the maximum intercide distance, we will get a prior on phi as 3 divided by d max and 3 divided by 3 divided by d max and 3 divided by d min. This should be d min. There's a typo there. All right. So that's the basic movement into the Bayesian geostatistical models. With regard to this slide or any of the previous slides, are there any questions? Because I'll then move on to Bayesian modeling with CAR models, so that those are like regionally aggregated data. Okay, so let me stop sharing this and go to the other aspects, which is also useful. Let's see if I can share that screen. Yeah, please feel free to ask questions because I'm not very good at monitoring the chat. Oh, sorry.
Yeah, in the morning, I mentioned that you just put it in the chat. So now we have.
Oh, I see. Yeah.
But
okay.
Feel free to ask any questions.
Yeah, just give me a second. For some reason, my share screen is not working very well. Okay, here you go. And let me just, okay. So until now, I have spoken pretty much about modeling for the for GIS data that are point reference, right? In one of the first examples in R that I showed you, though, it was not point reference, it was county level data, right? And in public health, depending upon what your field of area is, in public health in particular, or some of the biomedical population-based studies, economics, social sciences, it's very common not to get data at that high a resolution. And in public health particularly, it is not provided at such high resolution by design, because we want to make sure that that we reserve the confidentiality of, let's say, patients or individual subjects, and then so we don't want to go that far. So let's take a quick look at a different type of effect, right? So first, very quickly, I mean, just to recap, in general, let's imagine you have data that is coming from different spatial units. If you actually have no spatial association, then you will fit a linear regression like this, a multiple linear regression like this. The errors you will model like this. The mean function hierarchical comes in here. Again, right here, I'm not going to repeat this, priors, everything, just standard linear regression, right? Binary data, GLM, no random effects, right? Okay. Random effect models, the first thing you do is simply introduce random effects. So I'm not putting as SI here anymore. I am simply putting WI because these are regions. S essentially is a site. You could still put it as WS of SI. The notation here is typically saying that, let me just index the region or the county, let's say, if you want to just have a concrete imagination in your mind. YI could be lung cancer rates, MUI is the mean, X size could be certain risk factors. I denotes the county from where that rate is coming. And then, so therefore the spatial random effects are county level effects, and you're going to have some kind of structure here. Similarly here, for continuous data, for count data, you can do that for binary data as well. It's the same. You will introduce the random effects, and then this correlation matrix or covariance matrix rather is going to introduce dependence. Okay. Now, here comes where there's a pretty big departure from the geostatistical models, where you actually are thinking about a spatial process and so on. So let's look at the matrix. Let's look at a map of California or any other, maybe the United States, and that has been delineated by regions, counties, states, zip codes, okay, any administrative unit. From the map, you can actually extract from the GIS database the neighbors of a region. That's possible. In fact, in one of the programs I have put in the folder, which I will not unfortunately have time to go through, you will basically see how you can actually extract neighbors, adjacency matrices. You don't have to look at the map and manually go about finding neighbors. The program will do it for you. And so you have these entries, and what you actually look at is an adjacency matrix, okay? So the entries are ij. AII in an adjacency matrix is 0. And AIJ very typically could be 1 if ij share a common boundary. AIJ could be an inverse distance between the units if I and j are kind of defined as neighbors that way, that look at that. And then or it could be AIJ is 1 if the distance between two units I and j is less than or equal to k. Distance between units may mean distance between the center of the two counties. and Aij could be 1 for m nearest neighbors. So you see, this adjacency matrix does not need to be symmetric, first of all. For example, this last one, all right, is it could be that Aij is 1, but Aji is not 1. It's just because I is among the m nearest neighbors of j does not mean that j is among the m nearest neighbors of I. J could be surrounded by a lot of other points, so that are closer to it than I. But I may not have that many points. So it's easy to, put some points on a piece of paper and figure out when that can happen.
So if I understand, these are different ways of defining the matrix.
Correct. Absolutely. Absolutely. Exactly. Thank you. And then, this A tilde, what you do is you standardize the row by simply scaling each variable Aij by dividing each element by the number of by the sum of the row of the Aij. So in the binary setting, Ai plus will simply be the number of neighbors of I. The only thing common here is that if I and j are not neighbors, no matter how you have defined it, then Aij is usually taken to be 0. All right. Sometimes these elements are called weights. I don't know, perhaps helps interpret it better. Okay, now the car model is a bit more of a technical thing here, in the sense that it actually does not introduce dependence directly through a correlation or a covariance like we did for the spatial processes. There's a fundamental difference in this model. What it actually does is that it specifies conditional dependence or conditional independence. So you look at the Wi given all of the other effects, look at the normal distribution and say that it's a normal distribution, take a weighted average over its neighbors, maybe. Say, for example, if Aij was a binary adjacency matrix, for example, this would basically become the average number of neighbors, right? Or the average of the Ws, rather, I should say, over the neighbors of I. That would be the mean. And then the sigma squared, which is the variance, common population variance, would be divided also by the number of neighbors, so that you know that a region which has more neighbors will have more precision. And the joint density then can be shown through some, pretty neat, not too difficult, but fairly standard probability theory to be a multivariate normal distribution under a pretty generic assumption. So the precision or the inverse of this variance matrix, in fact, from this form has a very nice interpretation. For those of you who are familiar with graph theory or graphical models, will recognize this guy to be the Laplacian of the graph, which is used in computer science, statistics, data mining, a lot. Graphical data mining, I should say. Basically, what this means is that the precision, the inverse of the covariance, is scaled obviously by 1 over the sigma w squared. But then right here, you have a matrix D, which is simply the diagonal with the number of neighbors. If in the binary matrix setting, just imagine it's a binary matrix, it's easier. And the A is the adjacency matrix and rho is the correlation matrix. It doesn't have to be the binary adjacency matrix. It could be any adjacency matrix formed like anywhere like this, but then the diagonal of the D is the sum of the corresponding rows. Okay, as I said, the very usual specification is Ai is 0 and Aij is 1 if I and j are a neighbor to each other, and 0 if not. Now, what is
there
interesting
is a question on that?
Yeah, go ahead. Go ahead.
It's in the chat.
Oh, I see. I'll take a look. I'll take a look. No problem. Mauricio Cruz. Is the adjacency matrix usually defined in a discrete way, or is there a way to account for example, how long the boundaries are between neighboring counties or those kind of things? Excellent question. This is one of the major advantages of, oops, I'm sorry, I'm going the wrong way, of looking at this adjacency matrix. You see, if you can define your neighbors in any way you want. So if you have a geodetic software that actually gets you the, in fact, using a very powerful package in R called the raster package, you can actually do exactly what you're talking about. You can actually find out the distance of the boundaries or the length of the boundaries between two regions, and then have a threshold or cutoff. So just like the third bullet, except that instead of looking at distance between units, you replace this that the length of the boundary is greater than or equal to some threshold. And you can form the adjacency matrix like that. Now, you could even look at AIJ and make that as the distance between the distance of the not the distance, sorry, the length of the boundary between the two units. That can be done. Unlike in the previous setting where the correlation function had to be created from a covariance function which had to be positive definite, in this setting the adjacency matrix can be whatever you want it to be. Whatever you want it to be. But it will be an n by n matrix and its elements have to be defined. You define them in your way as needed in your research or in your application. Because in the end, the whole beauty, and I'm about to come to that, which will actually perhaps clarify the important technical underpinning to your question, Mauricio, is that if you form this d minus rho of A, and in a way that rho is bounded between the eigenvalues of a particular matrix, d to the power minus half A, d to the power minus half, in fact, in very generally, then this matrix is in fact going to be positive definite. And you will have a valid model. You will have a valid model. So you don't have to worry about the adjacency matrix. It doesn't matter what the adjacency matrix is. As long as from that adjacency matrix, you are actually calculating the D like this, and you're using this, that's the role of this rho parameter. You're using this rho parameter to remain within a particular bound, you will actually get a valid probability model. But if rho is equal to 1, in other words, you end up with D minus A, you clearly see it's a problem. Because if you multiply this matrix with a vector of 1s, you get 0. Why? Because D times vector of 1s gives you this, and that particular row of A times 1 also gets you the same thing. So that becomes a 0. So it's an easy consequence of matrix multiplication. Okay, let me take a quick look at a very important exercise in spatial learning or spatial data science as applied to biomedical applications, public health, and so on. So this was a project I had worked on many, many years ago, I think in the early 2000s. And I used to live in the state of Minnesota there, which is what you see up here. And each county reported infant mortality rates. This is the type of data that we get regularly, even from California for that matter. And you will have an intercept, and then you will have fixed effects, and then you will have a county-wise random effects, and so on. So you want to incorporate all of that. That can be done in the hierarchical model. Intercept, of course, is nothing more than a regression coefficient. Fixed effects are like predictors, risk factors, if you have them. So there will be a regression component there. And then the random effects that we have been talking about. Now, let's suppose that when you do this, this is called a car model. Okay, there is a role for the improper car as well. It actually smooths well, but I'm not going to talk about that too much because it gets into a lot of technical stuff. You can still fit an improper model, in fact. But if you fit the car model, right, you see this map below this other, the IID map. And what you see is a massive amount of clustering in the north for high infant mortality rates. So what we are actually looking at is that, let's say you have without covariates, You have an IID model, meaning you put in random effects, but they're not spatially correlated. You get a map like this. Then you include covariates. This map changes here. If you look at both these two maps, there's a fair bit of randomness, and it's not clear what the spatial clustering or the pattern is. What the CAR model does using this specification is helps you learn better about clustering from the data. Remember, we're in the business of learning from data, right? That's what machine learning is. That's what statistical learning is. That's what we all do. And so we are able to learn much better about these effects from the CAR model, because you get the pronounced clustering. And there's reason for this. You know, the northern part of the state, in fact, has much less access to healthcare. There is a large Native American population there who have been deprived of healthcare for different policies and so on historically. And there still is a lot of suffering there. And on the other hand, when you put the covariates into a CAR model, let's say if here in this particular case, in fact, one of the covariates was something that informed us about the economic status of the northern communities in Minnesota. You still get that, but it's kind of mellowed, right? Because the covariates are playing a role, just like in that previous point reference example I showed you, where the covariates can actually mitigate the pronounced effect of spatial random effects. Here too, the same thing can happen. So you have the Bissag, York, and Mollier model, which is a very famous model. And here you actually add on a random effect IID, but the CAR still comes here. And then similarly, you have other types of modifications, like modifying this using a weighted average of the random effects, and simply an identity matrix, an independent matrix. So that's why this 1 minus rho comes in, and you can work that out. Okay, so this is a completely different way of doing statistics using adjacency matrices and so on. I am not probably going to work on have time to do all the details about this, these two different types of models and how you can do that. If any of you are interested and you're in the UCLA campus, you can feel free to set up an appointment with me, come and see me. I'll be happy to discuss your problem in spatial statistics and help you out. In the last 15 minutes or so before we end, I want to show one demo of Bayesian modeling, of course. And let me actually, there are quite a few different files here. One is for big data models like nearest neighbor Gaussian processes. That might be a little bit more specialized. If you have really big data, like millions and millions of locations, then that's useful. We are really scaling up to 40 million, 50 million now with the advancements that have been made, not just in computing, very importantly in statistical modeling. Okay, because you cannot solve the big data problems in spatial statistics with only computer power. You still won't be able to, store and implement everything there. You absolutely need smart modeling. You need graphical models and other stuff to actually do dimension reduction. So let me actually come here. Let's see. How about this? Okay, what I'm going to do is, instead of running, which is going to take a while, for those of you who are in RStudio or in R, you can run this R markdown file that I have here in the folder. And, oh, actually, you can't see me. Sorry, hang on. I'm going to stop sharing and let me put on my, oh, is there another question? I see something in the chat. Oh, thank you. You're welcome, Mauricio. Let me actually, share screen again. Maybe I'll just share the desktop. That'll be easier. How do I share the desktop? Right here. Okay. I'm going to share the desktop and let me bring in this. So there's an R markdown file here. Oh, let me close the car model. Let's pick this up. And that actually generates this HTML file. So let's take a quick look at this. You should be able to run the R markdown file. It's fairly bare bones. You download the R markdown package and you use the render function to actually create the HTML file. That's how you do it. Of course, if you're in RStudio, I'm sure you'll just be pointing and clicking. I'm sure Casey knows how to do that and can tell you how to do that. Maybe he already has. So, in Colorado, we actually have a lot of temperature data analysis going on. Colorado presents, the UCAR and the NCAR, which the NCAR is the National Center for Atmospheric Research, and the UCAR, I think, stands for, I forget what the U stands for. But anyway, they are centers that that collect a lot of climate data, assimilate a lot of climate data, do a lot of data science.
That's a University Corporation for Atmospheric Science.
Thank you so much. Thank you so much. And there's a lot of climate analysis going on there. And one part of my work in spatial statistics definitely overlaps a lot with climate data science. So I have been involved with NCAR quite a bit. So there is a data set about temperature across Colorado, and you can download that here. I believe I've also placed it in the folder. So if ever, this is my former student, Andrew Finley's lab. If this link goes away, you will still be able to probably get that data. So you load that data. This is the first segment. You load that data. You can just copy and paste this. I don't think I'm going to go and do this because it'll just take up time. I would rather go through the different steps. And then, once you load the data, you can see. It's always good to see what the data is. And so you can look at the, you can use this LS, the list, basically listing of objects function with an empty argument, and it'll give you the data's different attributes. So you have coordinates, which gives you the point level locations. You have elevation, which will act for us as a predictor. We look at the prediction coordinates, the coordinates where we want to predict temperature, because we have not measured it there. At those locations where we want to predict, what is the elevation? We know that. We have included that. And then temperature is our dependent variable of interest, okay? So that's what we want to look at. So our goal is to create a complete prediction surface, right? Here's our main goal, to create a complete prediction surface of minimum spring temperature with associated estimates of uncertainty. And so we begin by loading the necessary packages. So we have SPBase, we have MBA, we have GOR, we have raster, we have leaflet, and we have SP. I think SP will already be loaded if you load at least SPBase and a couple of others, because there are prerequisites for that. But leaflet is a very interesting and nice way to look at dynamic visualization. So again, I won't have time to go through all the features. This is, as I said, we have a 1-quarter course. If you guys are interested, you can take it. If you're a UCLA student, it's free, right? I mean, you're already paying your tuition, so you can come and do that. But I don't have time to offer it every year. So here you have these different colors. Color codes, blue to red, and these are codes that are inbuilt. Don't worry about them. Don't mess with them too much. They look nice. That's why I put it. There's been some experimentation in terms of the visual aesthetics of these programs before we are presenting these to you. Then you have a base map, and then you have the region, the stemane terrain. There are different tiles that you can use. You can use ESRI world imagery. That's like Google Maps. And so you can get that as a satellite version. And then the terrain version is here. These are all inbuilt. You don't need separate files for this. Okay. So this is all part of the leaflet package. Then you create a palette. You create your base map. with different parameters. You can add a legend. You can play with these, the legends and so on. But what the leaflet package does is gives you a dynamic point here. So you see, this is the terrain view, and you can easily switch to the satellite view. These points that you see are the points where we have observed the temperatures, and we have color coded them. So as you can see, just no surprises whatsoever. Here is a part of the Rocky Mountain range, and so you can probably see, well, is it the Rockies? I suppose, yeah. Anyway, there's higher elevation here, and you can, in fact, look at the points that are giving you the temperature values, and they are much lower. They're in the negatives. The red is the higher temperature. And so right here, you have the red, which is more in the desert and the plains. Okay. So, oh, by the way, if you want to switch to a satellite, right here, this is a satellite image. And then here's a terrain image. So it's a matter of preference, what you want to put in. You can switch between the two. You can save both of them if you're publishing an article. If you're in an electronic journal that allows dynamic pictures, you can just include this picture in an electronic journal. Okay, then you can fit a non-spatial regression model, and you can just simply use latitude and longitude. And this will essentially give you a value of the residuals from which you will get an idea about your residual variance. Not going to spend too much time on that. We come back to reprojecting the geographic coordinate to a UTM coordinate system. There is one important reason why you want to do this. The reason is that, there's positive definite functions that I talked about, those correlation functions. Very strictly speaking, mathematically, they are valid only for Euclidean distances. But what happens in places like Colorado or even in the continental US, Canada, the curvature of the Earth is not so much that if you use the geodetic distance or the great circle distance, they will still work. Many years ago, I think around 2001 or so, I have a paper in a journal called Biometrics, where we talked about these geodetic distances and their effect on spatial modeling. And you can read that paper. And my conclusion was that we don't have to obsess about that. But if you're looking at, let's say, the entire Pacific Ocean, and you're looking at sea surface temperature, right from Japan to the US, and you want to actually build a correlation matrix there, then the curvature of the Earth could produce discrepancies, and you may get some errors, perhaps even in terms of positive definiteness, or if not that, at least your result is going to be a little different. So UTM projections are easy to compute right here. Project the coordinates so you basically pass these coordinates to the SP package using a spatial points object. That's basically what it does. And then you convert it back. So here SP transform does that, and then you convert it back to just the simple coordinates that you can use for the statistical analysis packages. A lot of statistics and data science interplay in R is about going back and forth in terms of data formats for what you need it to be. Maybe Python has that advantage. It's a little simpler in that regard. But let's take a look at the regression model residuals. We also look at the Dmax, as I said, which was the maximum intersite distance among the points in Colorado, right here. That's very easy to calculate, the Dmax. You can just simply calculate the max of the I-dist as a function that calculates the intersite distance. I stands for inter. And so if you have the UTM coordinates, coords.utm right here, you could easily calculate that. And then again, you calculate a variogram. I'm not going to go through this in detail because we have already seen that. But look, after you look at the variograms, you basically see the increase here. Now, it does dip later on. This is what we call a sign of non-stationarity. But if your domain is not very much beyond this, it doesn't matter. A stationary approximation will still be pretty okay for your predictions and your analysis. I don't think it is going to change particularly predictions are quite robust. So you do that, you calculate those variograms, and then you can calculate your interpolated surface, right? And here you can again use your raster image to actually overlay the interpolated surface. This is the deterministic MBA interpolation, multivariate B-spline approximation. And you can click on that, or you can click on the residuals, the satellite version or the terrain version, whatever you want. And this is where the interpolation is being taken care of. And you can do that for the residuals, or you can do that for the variable. So again, you're here, you have the satellite region, you have the terrain. If you come to the residuals, it plots that. And you can repeat that with your the value of the variable as well. Then you're going to fit some spatial regression models. This is all the, here is a very useful one. It's in the SP Bayes package. These are all being done now in Bayesian settings, okay? So the difference between the previous R session and this one is that we're now fitting Bayesian models. Again, it's already 4 o'clock. TV, can I get about 5 more minutes?
Sure.
Okay. So what happens here is that you see here, I'm actually fixing a couple of parameters from the variogram. For example, the phi, which is a decay parameter, I fix it at 3/200 by looking at the variogram. How do I do that? If you come to the variogram, you see it's at about 200. Here is the residual. So the residual at around 200 is where things start flattening out. Now, you might say, no, Dr. Banerjee, it's not 200, it should be 250 or 300. That's fine. You choose what you want. The alpha is a ratio between the nugget and the spatial variance. And so that one on the residual scale for the variogram is right here. You know, it's about, the nugget is about 1, whereas the variance, the total variance or the spatial variance is even over 12. So you could do about 1/12, okay? And then you can build what is called an exact sampler. It doesn't need MCMC. It does a little bit of distribution theory and gives you samples from the exact Bayesian distribution, the posterior distribution. So then you have the trace plots. These are obviously not MCMC, so the image, these are direct samplers, so they're just awesome. But if you want priors on those guys, now we are fitting the full Bayesian model, a full hierarchical model. This is how it's specified. You now have priors, and you also need, for your updates of the range parameter and these variance parameters, you need metropolis updates. So that means you need tuning parameters, and these usually you need to experiment a little bit, but you can get what is called the Hessian matrix from a maximum likelihood estimate and see their diagonal values and plug them in. That's usually a good way to start. And then, you know, you run that, and here you have the trace plots. This is, everything is unknown. It's more flexible, but it will take a little bit of time to converge. And then after convergence, you can take the samples, and here is your trace plot. They suddenly have some autocorrelation, but they're not too bad. Look, particularly the tau square, it knows it's where it started, it knows where it's going. The phi kind of is uniformly distributed. So there's a little bit of learning there as well, but not probably a lot. Sigma square is like this. Then what you do is you actually recover the beta and the W. And that's actually easily done using this SP recover function that gives you the posterior sample of the beta and the W. So the method is to first sample the posterior, the covariance parameters, then come to the regression coefficients and the spatial random effects. That's much more efficient. And then, you know, you can get to the quantiles, and you can look at your, you can round them up, and you can look at your predictors, and if anything is significant or not. If 0 is included in the credible intervals, then that means that nothing is significant over and above the spatial effect. And then, you can look at quantiles, and then you can do the predictions. So the prediction actually, again, you can look at this quite closely, but it pretty much is the same structure here. You first get the predictions using this spatial effects and extracting them, and then using the surface plot to plot them. So again, you get the random effects and regression residuals, and you can plot them right here. You can do this for different models. And so here is SPLM M1, which basically means a full model with everything has a prior, whereas here it's you have actually fixed a few parameters. So you see they're a little different. But overall the pattern is being captured fairly well. And then here are your residuals, and you see a lot of spatial structure here, because we haven't until here put in the elevation, right? So what we'll do now is we'll put in the elevation into the mean, and then we do repeat the analysis. I'm not going through this side by side. And then you get your spatial random effects again, and right here you see the random effects, and there's still a little bit of structure, but it's way more mellow than the other models. So anyway, I'm going to go through here, and then finally you have the prediction for temperature. You could also look at the interval widths, and you see the interval widths are sort of prediction interval widths. So we are getting a lot more prediction. The widths are lower because we have data here, but as you go towards the edges, we didn't have much data, so that's why they get wider. And then so on. Finally, you can do model comparison using DIC and so on. So you can, typically see that using the model 2, which has the full spatial model, is way smaller DIC than the first model. So I think I have to stop here because it's almost it's past 4, and I've only managed to give you a tip of the iceberg, so to speak, in terms of the power of R and the power of Bayesian modeling for your GIS data. But I hope it will stoke your curiosity to look further, plunge deeper into this area. And hopefully, if you're working on interesting scientific problems, you will be able to make use of some of these functions and perform Bayesian analysis to work out to get better models and do better data science overall. So thank you. On that note, I'm going to stop.
Any questions from the audience?
I have a quick question. How much do you think the choice of covariance function matters in practice? And how fussy are you with your choice of the covariance function in geostatistical models?
Yeah, good question. If the primary objective is for spatial interpolation, like getting the random effects and the predicted surface and getting predictions fairly right, it's not a big deal at all. In fact, there's theory. Forget about the empirical evidence. There is also theory strongly supporting that misspecified covariance functions will still do pretty well in Gaussian processes. And there is a reason for that. I mean, I can't get into the mathematics of it, but just to give you a very brief reason, the Gaussian process is an interpolator. So that means if you actually take a look at, so you fit the model using a GP. Now, if you actually take a location where you actually had the data, the fitted data will exactly coincide with the observed data. And because the GP is an interpolator, it has that flexibility to capture the surface being suggested by the data and learn from it. So the covariance function almost plays a kind of like sort of like what in nowadays in deep learning will be called just a tuner. And you can, it's pretty flexible in tuning. You will be surprised how good it is. So I don't obsess it as much. There are certain specialized areas in statistics that I have worked on, one of which is called wombling. You can Google me or you can Google wombling and you will find it. And that actually talks about inferring on the smoothness of the spatial process. So if you're interested in a rate of change on the process, then you really need some covariance functions that are specialized to allow you to do that. And there the matters become a little more complicated.
Awesome, thanks.
Thank you.
Any other question? Anybody else? If not, let's thank Professor Banerjee for the excellent presentation, and that's full of knowledge. Hope you will make use of it, and please reach out to him if you have any research going on in this area.
Yeah, sure. And one thing, I know Casey will probably be gone, and I don't think I can do it in the near future, but TV, just so you know, if in future, if you think there is enough interest, I can do a whole day on this, only on the spatial part. But that will require a lot more planning because my schedule is really tight. I don't think I can do it this year at all. But I am available. I'm interested in interacting with folks.
Sure.
And I think that it could be done jointly with the Institute of the Environment as well.
Yeah, I will take that offer at some point. Yeah.
Thank you.
Thank you very much. Thank you all for joining. And again, thank you to Professor Banerjee. And thanks to Casey for proposing this workshop. So it's been excellent.
Yeah,
so
it wouldn't have been happen without Casey. Casey's the main organizer and the coordinator.
Yes.
Nor you both though. So yeah, thanks so much. This has been great.
Thank you, TV. Thank you for organizing this. It was a pleasure.
Thank you.